I'm
Yeah, you host now.
yeah, I'm recording right now. Andres, you can start.
Awesome. So, first of all, I just want to say thank you guys for coming. I know Friday afternoon, the week of reading days is a little harsh to come, but I really appreciate you guys coming. Also, I want to just give a big shout out to the rest of the people that came together to help make this PowerPoint. All the other SIs are rock stars, and I really have appreciated working with them. Let's go ahead and get started. So I'm going to be going over chapters 41 through 43. We'll have some questions along the way, and if you guys have any questions in chat, please, one of the other SIs is going to, very graciously answer any doubts you have. So let's go ahead and get started. So I really wanted to focus on experiments, just because they're very important. They tell us how we got to where we are, right? So one of the keynote experiments that we saw was that there was this phenomenon of bacterial conjugation with the Streptococcus pneumonia, right? And we want to think about the fact that with Streptococcus, there's two strains, right? There's these rough strains that basically look like little candy dots, right? And then we have these smooth strains that have this more kind of irregular shape to them. They kind of have this smooth surface to them. So we've actually, through bacterial conjugation, we saw that this rough strain is actually able to turn into the smooth strain, right? So when that happened, right, in 1944, Avery Oswald, set out to go ahead and try to figure out what is the information basis of how this phenotype change occurs. So, at the time, people realized that there is such thing as bacterial conjugation. And what we want to think about with bacterial conjugation is that bacteria will be roaming around. Sometimes they'll come across a dead bacteria. And these bacteria are phenomenal machines at, being able to cannibalize, use what's around them, right? So one of the things that they can do is that they can just chew up on the dead bacteria for just building blocks. But another thing that can happen is that sometimes it's able to take in, genetic information and express a new phenotype that it didn't have before. So how did this happen, right? So this was a big question for Avery Oswald. He really wanted to understand what passes the information throughout, right? So here, as you mentioned, right, it's this mixture of the rough strain with a cell-free extract of the smooth strain that allows the rough strain to now express the smooth strain like phenotype, right? And we didn't really understand how this transformation came about because we didn't understand the molecule, right, that was being used to confer the information, right? So here, and lastly, I just wanted to say a little note about, the smooth appearance is because of a polysaccharide capsule that helps it protect it. So that's why the smooth strain ends up being pathogenic, right? The fact that it has this extra protection stops it from being kind of degraded by immune systems. But let's go ahead and think about how this works, right? So what we first do is that we grab a strain of a pathogenic smooth strain of the Streptococcus pneumonia bacteria. Then we go ahead and treat it, we just go ahead and heat kill it. So what this will do is that this will create a cell-free extract. So just imagine all the nice little contents inside of the bacteria just lysed out. And with this, right, we have all the components of this bacteria, right, just around in the media floating about. And this is great, right, because we're able to treat it with different types of breakdown proteins, right? So we can treat it with proteinases, ribonucleases, deoxyribonucleases to kind of figure out, see what is in this cell-free extract, right? So just very quickly, we want to understand that proteinases are going to break down any protein in there. Ribonucleases are just going to break down RNA, and deoxyribonucleases will break down DNA, right? Importantly, the actions of proteinases won't have effect on RNA and DNA, and so on and so forth. So what Avery found, right, was that when he went ahead and treated the cell, when he treated it with proteinases, with the cell-free extract, and then mixed it with a rough non-pathogenic strain of Streptococcus pneumonia, nothing happened, right? The rough strain is still able to turn into the smooth strain. So then he treated it with ribonucleases, and we saw that the rough strain was still able to turn into the smooth strain bacteria. But then when we treated the cell-free extract and deoxyribonucleases, we no longer see this the rough strain becoming the smooth strain, right? So this kind of gave us a hint or a direction to follow as far as what is the genetic material, right? So this is a great experiment that showed us, again, that for us in our planet, DNA is going to be the information-rich molecule that's going to confer, genetic information, right? So let's go ahead and think about this question. So here I'm asking, let's say you're in Mars and you're trying to figure out what the genetic information molecule is for these Martians. Let's say you find two strains, right? One that's pathogenic to the Martians and one that's not. And you want to figure out what the bacterial conjugation is, right? So you go ahead, you treat it with the same experimental setup that Avery Oswald did, and you get the following results right here. So you see that it's insensitive to proteinases, it's sensitive to ribonucleases, and it's insensitive to deoxyribonucleases. So here I want to ask, right, what is the biological molecule for these Martians that's carrying information based off these, based off this reading? So let me check the chat really quick. Oh, awesome, great. So a lot of RNA, so of course here, right here, because it's sensitive, it was sensitive to RNA, we would just go ahead and say that here, RNA is going to be the genetic information carrying molecule for this weird Martian strain. Awesome. So we also want to be aware about the fact that we have really many conformations of DNA, but we really want to be aware of three, right? The first one is going to be the B DNA conformation, right? This is going to be our typical helix, right? So here we have a picture of it to the right, the middle helix that we have here in this picture. We want to know that it's right-handed, that it has 10.5 bases per turn, right? And here we see that all the bases are lying perpendicular, right? So you're lying perpendicular to the axis. Then we also have this other form, which is called aDNA. This is also right-handed, but this is usually seen in dehydrated conformations of DNA. Also, we also see that this a double-stranded RNA and RNA-DNA hybrids will take on this aDNA conformation. And lastly, we have this zDNA that's just going to make a very small amount of our DNA molecules, right? And it's, in this case, it's going to be left-handed. So let's go ahead and look at this question. So here I asked that an mRNA can pair with an anti-parallel mRNA to form a double helix RNA. This double helical structure would most likely match which of the following forms of DNA? So if you guys could just put it in the chat. Awesome. Yeah, so it's going to match aDNA, right? Because again, we're going to have, we're going to have aDNA when we have these RNA or RNA-DNA hybrids. Awesome. So here we also want to be aware of these four important enzymes, right? These are going to be enzymes that allow us to cut stuff away. And in the case of DNase ligase, bind DNA molecules together. So here for endonuclease, we just want to think of it as something that breaks the DNA or RNA within a segment. I have some pictures to help explain this, right? For an exonuclease, we're going to see that it's going to take a chunk out of one of the strands of DNA. For an exonuclease, these are just going to go ahead and cut at the ends of some sort of DNA molecule, right? And again, DNA ligase is just going to ligate together two DNA molecules so that we can, create a phosphodiester bond. So here I have a couple of pictures, right? So up here at the top, we have this cute little GIF, right? And here's DNA ligase. And we just want to think of this as being the glue that kind of is able to ligate, phosphodiester bonds together. Down here, we have, a little image representing the actions of endonucleases and exonucleases, right? So in the case over here, we see that endonucleases are going to do mostly just internal cuts, and exonucleases are going to cut from the 3' end or the 5' end. And lastly, here's just a picture of an exonuclease, right? So here, in this case, it's one of the DNA repair ones, right? So here, we're going to have this section that gets removed. So it's just a section of one of the strands that gets removed. And, typically that's done for some sort of repair mechanism. But we just want to be aware of these four different enzymes and the fact that they're different, right? They're used in different situations. And yeah. So now let's go into 42, right? So in 42, we also learned about another experiment, Mercantile and Stahl. So this experiment was really interested in finding out that which actual type of DNA replication did we have, right? At the time, there were 3 modes of replication. There was this conservative mode, which meant that the strand of DNA simply duplicated or just cloned itself to have another one, but the original one was conserved. A semi-conservative, where we have one strand serve as a template, and then either strand can serve as a template, and then we form a hybrid molecule, right? And then we also have non-conservative, which is basically meant that DNA, when it was replicated, just was built from the ground up each and every time, right? So At the time, we thought that there were these three possible methods, and this Matheson-Stahl experiment helped us figure out which of these modes of replication are seen in our cells, right? So to do this, what we did is that we grabbed a bunch of E. coli bacteria and we grew them in media that had a heavier isotope of nitrogen, right? In this case, nitrogen 15. Let's just remember that the normal isotope of nitrogen is nitrogen 14. So when we grow it in this media, as we remember from exam 1, right? All this nitrogen is these nitrogen 15 isotopes will be taken in and they'll be incorporated into the ribose sugar, ribose sugars that are going to be holding up our nucleotide bases, right? So this is a way for us to kind of make that DNA be a little heavier. And this, again, set that, boom. And then what we did is that we go ahead and when we let these just divide for we split this between a first generation and second generation. And we put those into these cesium chloride gradients, right? So in this first picture up here, there was no replication that was allowed, right? And then it was put in the cesium chloride gradient. And what happens is that as we see when it gets centrifuged, most of the DNA ends up being at the bottom. So again, just so that we orient ourselves with this figure, right? The lightest molecules are going to be closer to the top of the center of the tube, and the heavier molecules are going to be closer to the bottom. So what we saw here is that we went ahead and again, we did replication. When we did replication, we showed that there was this, when we had a single replication event, right? We saw that there was now this hybrid band of DNA, right? Where it was just kind of in the middle. It wasn't all the way down where the heavy DNA part was, but it was kind of in the middle. So at this point, we can go ahead and cross out the conservative, right? It seems like there's going to be some sort of hybrid that's being formed in this case. So after this, we go ahead and let it go through another round of replication. And when that happened, right, now we see that the two original strands are incorporated here, but now we have two new DNA molecules that are just going to be nitrogen 14 on their own. So as we see here, what this ended up showing us is that here we ended up having semi-conservative replication, because as this replicates, right, our hybrid DNA stays at this part of the gradient, but our new lighter DNA, that's nitrogen 14 DNA, is not going to be able to rise to the top. So we just want to be aware of what the ramifications of this experiment and how to interpret, where our bands show up. So let's say that instead, right, in growing the media, right, instead of growing in nitrogen 15, we grow it in a nitrogen 13 isotope. What would change in the Matheson-Stahl experiment under these conditions, right? So here, right, what we want to think about is essentially the experiment would just be reversed, where instead of seeing the nitrogen 15 isotope at the bottom of the test tube, we would see the nitrogen 13 isotope at the top of the test tube. And when we go ahead and have DNA replication, again, we're still on Earth, so it's still going to be semi-conservative. So instead, the hybrid band is going to show now below the nitrogen 13 band. So I know it's a little complex to kind of explain this over chat, but I just wanted to go ahead and throw this question at you. And we in this case, we would still say that, DNA replication is still semi-conservative. So now we also have a lot of mechanisms to prevent error when we go ahead and do DNA replication, right? So we want to be aware of these three. So I have some images that we want to think about. So in the case of pre-synthetic error control, we want to think about the fact that conformation is going to serve as this pre-synthetic error control. So in this picture, right, we see that the catalytic site has the ideal conformation for normal Cricket and Watson base pairing, where we see a C pairing up with a G and a T with an A, right? But we see that when we have other abnormal pairing, this is going to kind of not be able to fit into our catalytic site. So this is a way for us to have pre-synthetic error control. Another thing that we're able to do is that we're actually able to go ahead and have post-synthetic error control or proofreading. So what this basically means is that we can go ahead and have some sort of, let's say we have a incorporation of a nucleotide base into DNA. What's able to happen is that the polymerase itself has this little exonuclease activity that it's able to go ahead, back up, cut out the base that, the wrong base that we had there, and then it's able to reshift itself so it can go ahead and try again to put the correct base in that case, right? So this is a way for us, for the RNA to kind of fix the error as soon as it happens. And in this case, they would serve as the example of post-synthetic error control. We can also have, mismatch repair, right? So after replication is done, after the polymerase is, dissociated, and we already have a DNA strand, right? As one of my SIs will go ahead and talk a little bit more about later, we're going to see that these DNA damages can go ahead and be fixed through, either nucleotide excision repair or base excision repair, right? But importantly, this allows us to, just general repair allows us to go ahead and fix any error that was unable to be fixed by the pre-synthetic error control or the post-synthetic error control. So now we want to be aware of the replication. We want to be aware of this, the replication forks in prokaryotes and eukaryotes, right? So one of the biggest problems that we have is that for the leading strand, right, we always want to remember that we have synthesis that's 5' to 3'. But for our leading strand, that's able to be the case, right? Our leading strand here is 3' over here. Over here we have the 5', right? So for our leading strand, we're able to have synthesis continuously. But for our lagging strand, what we see now is that we need to form these constantly be forming these Okazaki fragments so that we can go ahead and form the DNA. Because again, DNA replication always occurs 5' to 3', right? We never see 3' to 5'. So this is something that, both systems have to overcome. So in the case of prokaryotes, what's first going to happen is that we're going to have binding of DNA A at the OREC, right? So the OREC is going to be this cis element in the DNA that the DNA A is able to bind to. And what this will do is that this will go ahead and kind of promote the polymerization of DNA A such that we form this figure over here. And this will lead to a stress in the DO element, right? So this DO element is really, it's really easy to like melt, right? So what happens here is that all these DNA A's are just going to go ahead and basically open up the DNA and allow the transcription machinery to land here and be able to replicate, right? So again, here we know another key star here is going to be our DNA B, right? So DNA B is going to go ahead and migrate in the 5' to 3', right? And the binding of DNA B is going to commit the cell to the replication and division, right? So after this, we're going to have the release. These, of course, DNA B is going to be brought on by these chaperones called DNA C. And after that goes ahead and leaves, we can go ahead and start assembling we can start assembling our transcription machinery on here. So here for the elongation, what we're going to see is, I'll talk a little bit more in depth about the different units of the RNA polymerase, the DNA polymerase in prokaryotes. But what we want to realize is that here, it's the biggest components that we want to think about are going to be, of course, these core that are going to be helping this. We're also going to have a clamp that we need to load on. DNA B is going to serve as the helicase, and we're going to have a clamp loader that's going to serve to load on these clamps so that we're able to have the replication of this DNA, right? So again, in the case for the leading strand, we can have continuous synthesis. So as soon as we put a clamp down, we can just have continuous synthesis until we're done. But for the leading strand, we have to play this juggling game, right? So what's first going to happen is that up here, where the DNA B is going ahead and unwinding the DNA, what we're going to see here is that there's going to be a bunch of proteins kind of coating this and protecting it from DNases, right? But what we're going to see here is that this molecule called primase is going to bind, and what it will do is that it will put a short RNA primer onto our DNA, right? Once this primer is on, clamp loader is able to recognize that it's a site that it can go ahead and load a clamp onto it. So here in this kind of middle picture, we see that the clamp kind of bends itself forward. Clamp loader bends itself forward, puts the clamp down, and then we're able to just have the clamp on there. And then after that, we're able to just go ahead with the addition of the core enzyme, right? We're able to go ahead and have synthesis all the way up to the next Okazaki fragment. And then we would just have this clamp disassociate again. And again, for the lagging strand, this has to be a constant thing where we constantly have to be putting new clamps down on new primers that are put by primase. So let's just go ahead and look at this diagram here. So this orient ourselves, right? So here we want to recognize that here would be where the origin of replication was. Again, we're going 5' to 3' for DNA replication. Over here, this molecule we want to recognize as being the helicase. Here we're going to have primase laying down a primer, right? So it's going to lay down this short little primer. Then we're going to have, in this case, so here we're going to have a DNA polymerase. Here it says DNA polymerase 3, right? So here we're going to have this DNA polymerase 3, and it's going to start DNA synthesis from the 3' end to the from the 3' end, right? So here again, we're going 5' to 3'. Then in the lagging strand, we're going to have, of course, ligase kind of bind together these little nicks in the phosphodiester bond that we made. And of course, DNA pol 1 is going to replace the RNA primer with a nucleotide, right? So we just want to be aware of all this. And again, here for DNA pol 3, we can just have continual synthesis of the leading strand. Also, when we go ahead and have DNA ligase, let's just remember that it's a little different in prokaryotes versus eukaryotes, right? So in the case of prokaryotes, we're going to be using NAD+ as our nucleotide source to go ahead and cause this ligation to happen. But in eukaryotes, we're going to be using ATP, right? So just a little difference in the DNA ligase, but it's just something to be aware about. Then we just want to be aware that we're going to be showing that eukaryotic polymerase. So here we have a bunch of other enzymes, and these enzymes can a bunch of other proteins, right? And these are going to be very analogous to the ones that we saw for prokaryotes, right? So in the case of DNA, here in the core promoter is going to break its actions with the leading and lagging strands. So for the leading strand, we're going to have DNA pol E. Go ahead and do that, right? And here for DNA pol delta, we're going to have, it's going to be the one that's going to be in charge of doing DNA replication for the lagging strand. Of course, here for our primase, it's going to be DNA pol A primase, right? Pol alpha, this is going to be the complex that's going to go ahead and ligate our strand, right? Instead of having the DNA B helicase, we're going to have NCM. RF is going to be now the clamp loader. The PCNA is going to be our clamp, right? The ARPA is going to be the same as the single strand binding protein that we saw for the lagging strand. DNA ligase is going to be the same. And the last thing is that FEN1 is going to be the one that's responsible for being able to remove the primer. So here's a little chart. I'm not going to go over this chart, but this is just a quick comparison between eukaryotes and prokaryotes. So you guys have these lectures, so you guys are able to maybe review this on your own. I would recommend doing a chart like this, just because kind of really helps you see the differences in a one snapshot overview. But let's go ahead and now talk about lecture 43. So here we're switching on to prokaryote transcription and gene regulation. So here, right, we just want to overview transcription a little bit, right? So this is going to we just want to remember that this is going to be a DNA template dependent process, right? We can't have this transcription unless we have a DNA template. This is going to form the synthesis of RNA. This is going to be catalyzed by RNA polymerase. And of course, this transcription is going to begin at a promoter region. In this case, this cyan color shown in this diagram below, and a terminator region. In this case, this light, this baby blue kind of motif at the end, right? So both of these are going to be cis-acting elements that are going to be recognized by either RNA polymerase or other factors that bring RNA polymerase to the promoter. So here again, we have this difference, right, between trans-acting factors and cis-acting factors. So what we want to think about trans-acting factors, or really what we want to think about is that cis-acting factors are going to be DNA elements, right? They're going to be DNA sequence elements, and these will go ahead and have recognition sets that will be found by trans-acting factors, which are usually protein motifs, right? These are going to be things like your leucine zippers, like your helix, turn helix, like your helix, loop helix. So these all are going to be trans-acting factors that are going to go and interact with cis-acting elements, which for the most part, again, are going to be DNA sequences or promoters. Here, we also want to be aware that ribonucleic acid switches things up just a bit, where instead of having thymine, it's going to use uracil, right? So that's going to be the only difference. Thymine is really going to be, thymine is going to be replaced with uracil. Again, just like we saw with DNA, we're going to be reading this 5' to 3'. We have this ribophosphate backbone, right? And here, right, again, as you mentioned earlier before, these DNA RNA hybrids are going to be very similar to aDNA, right? So now we want to be aware of the types of RNA, the classes of RNAs that we're going to see, right? There's going to be really three that we want to think about. Messenger RNAs, so these are going to be important in coding the primary amino acid sequence for a protein. Then we're going to have these transfer RNAs, as you guys will see in the translation lectures. These are very important for us to bring charged amino acids, it's very important for us to use charged tRNAs for translation. And of course, we're going to have ribosomal RNAs, which are going to be important catalytic sites for the ribosomes to go ahead and create proteins. So now we just want to be aware of some terminology that gets thrown around when we're talking about this, right? So the first is, of course, again, we are going to start transcription of the promoter. It's going to end at the terminator. Once we go ahead and have a mRNA transcript, we're going to call this the primary transcript. In this case, when we go ahead and have transcription, this is always going to start at the +1 site. There is no 0, right? And anything that is in the 5' end is upstream, right? So anything that's anything that's going to be 5' to the +1 element is going to be upstream, and they're going to be shown in negative numbers. And anything that's 3' of the start site, right, is going to be referred to as downstream, and these are now going to be shown by positive numbers, right? Also, things that are going to be proximal are just going to be close to the start site, and things that are distal are going to be more far away. So here we want to think about primary transcripts, right? So again, let's think a little bit about the differences between prokaryotes and eukaryotes. For prokaryotes, they don't have a nucleus, right? So for prokaryotes, right, transcription and translation both happen in the same compartment, but that's not the case with eukaryotes, right? As you guys will see, there's extensive processing that needs to happen with eukaryotes. But what we want to think about, right, is the fact that this primary transcript, right, in prokaryotes is going to be essentially the mRNA transcript that will be read by ribosomes. Importantly, right, the DNA coding strand and the RNA primary transcript are going to be the reverse complements of the DNA template, right? So in this case, we see this example down here where DNA, we have DNA coding strand, right? And we see that the coding strand and the transcript are both mirror images of this template that we have here. So now let's go ahead and kind of put this to the test. So here I'm asking, let's say that you're given the following template strand, right? So it reads 5' to 3'. Which of these would serve, which of these would be the correct, what would be the correct mRNA that would be formed from it? So again, this is the template strand. So which of these options here would end up being the correct mRNA that would be produced? All right, perfect, right? So guys, so here, what we want to remember, right, is that we're going 5' to 3', right? And this is going to be 5' to 3'. So what we want to do is that we want to understand that the beginning part of the 3' end is going to be matched with the beginning part of the 5' end, right? So here what we want is that we want something that would be the inverse of A. In this case, it would be U, right? Then we would have CC. Then we would go ahead and have CC, right? And then we would have the rest of this. So here, again, we want to go 5' to 3'. So in this case, if the 5' is at the end of the 3' here, we would have 2 T's, right? So what we would see is that, again, this would be RNA, right? So what we would see is that the answer would be D, right? Because here, this one is going anti-parallel, right? So again, if we will go ahead and kind of graph D and just kind of match it here, you'll see how these strands end up being parallel, right? So again, just remember that we always want to read this 5' to 3', and the complementary strand should always read, the same 5' to 3', but it would be inverted. So now in prokaryotic promoters, again, promoters are going to serve as these 6 acting elements. And importantly for prokaryotes, we're going to have these consensus sequences that are going to be seen throughout the genome, right? So there's going to be this -10 element, right? So again, this is going to be upstream of the promoter region, and here we're going to have a -35 region. And we just want to be aware. You just want to be have these kind of motifs memorized. Where at the -35 we see this TTGAC, and at the -10 we see this TAAT, right? Or this TATAT box. So again, this transcription most often starts at the +1, and this most often is a purine element, and often it's within the sequence CAT, right? So let's go ahead and look at this example, right? So in this case, which of the following would be the best promoter, right? So let me check the chat. I'm gonna go over the last question. Yes. So again, here, what we want to think about. Actually, let me see. Can I annotate? Yes. All right, I can annotate. Right. So here, what we want to remember, right, is that this is going to match 3' to 5', right? So in this case, what we want is the transcript that here all the options are given to us like backwards, right? So here, if we look at D, right? For D, we have UU at the 5' end. So here we have U, right? And then we continue down. Here would be A, here would be another U. So here, right, we see how D basically matches this template strand, right? So that's why D ends up being the correct answer here. Again, what we want to remember is that this is going to be the mirror copy of it, but it's going to read in a different direction.
Wouldn't that be B because like you're reading from the 5' end and the question orients yourself on the like B orients itself on the 5' end, like 5' to 3'?
Oh, you know what? I see that. Yeah, no, you're absolutely right. The correct answer should actually be B. So yeah, that was my mistake, guys. My bad. So yeah, so here the correct answer should be B. Sorry about the confusion. You always want it to be 5'3" as the other person mentioned. Awesome. So here, you know.
Wait, so can you explain again what you do when you put it under? Like I see that you switch so it should be like, for example, B. Okay, no, never mind. I see what I mean. It's anti-parallel too. Okay.
Yeah.
Just as it would be to the template strand.
Yeah. Awesome. Yeah. So sorry about the confusion, guys. So, you know, moving. Oh, let me go ahead and erase this.
I have a question.
Go ahead.
So I guess I'm just getting confused on the last one. When you have T to mRNA, it's A, correctly? For some reason, I
Yes.
just wanted to correlate that with U. I don't know why.
No, so we would switch in mRNA any if we see a T in DNA, then it would be switched. You know, the complementary strand would be an A in mRNA. But if we have like an A in DNA, then it would be switched to U.
Okay, that's where I guess I'm getting confused.
Yeah, no worries, right? So again, we want to recognize these, you know, these two important consensus sequences in prokaryotes, right? And when we do that, so here for my next question, I was asking, right, which of these would serve as the best promoter? All right, perfect, guys. So here, you know, we can just quickly go back and remember that, you know, this is important for these sequences to be on the coding strand, right? So what we're looking here is for the one that matches the coding strand. So in this case, right? Right, so for the one, so again here, what we want to see. Perfect, right? So again, what we're thinking about is again the coding strand, right? So even though we have a perfect consent, we kind of have a perfect one in B, right? The correct answer would actually be A, right? Because again, what we're looking for, these consensus sequences, we want it to be in the coding strand. So here, the A, this is a little bit off, but it's still it would still be somewhat recognized by someone recognized as a promoter. Awesome. So now we want to go over to genes and open reading frames, right? So these are going to be, you know, units of heredity that function in the genome, right? Most are going to encode information that are needed for proteins, right? And importantly, right, this open reading frame is going to be a sequence that encodes the primary sequence of a protein, right? Importantly, in bacterial genomes, we're going to have operons, right? So here, what we want to realize is that, you know, in these genomes, we can produce polycistronic mRNA transcripts. So what this means is that it's just basically one giant transcript with a bunch of proteins all there. Additionally, right, we can have constitutive promoters that are actively initiating transcription. And of course, you know, the similarity to the sequence, to the consensus sequence is going to determine how strongly that promoter is at bringing in RNA polymerase. So just breaking down RNA polymerase. So again, in prokaryotes, what we want to think about is that this is going to be this is going to cause, we always want to have the formation of the hollow enzyme before we begin transcription. This hollow enzyme is going to be composed of these 6 little units that we want to be aware about, right? It's going to be composed of 2 alphas, right? One sigma subunit, which is going to serve to recognize the promoter, 2 alpha subunits that are going to be essential for the enzyme to assemble onto the DNA, and of course a beta and beta prime that are going to serve as the catalytic core of it. And omega is just going to serve as and omega, right, is going to serve as just a unit of structural stability. Importantly, when we go ahead and initiate transcription, right, we're going to have the hollow enzyme now become the core enzyme. So we're just going to unbind from sigma, and then we're going to have transcription continue. So in initiation, right, let's just remember that for initiation and DNA replication, we needed to have an RNA primer. But in the case of making a mRNA, right, we already have the primer, right? So in that case, we don't, this can be considered as something that's primer independent. So as soon as the hollow enzyme binds to the promoter, right, we see that there's this now open complex, right? And this now will allow the hollow enzyme to go ahead and start transcribing the DNA. So now that we have elongation, right, as soon as the sigma factor dissociates, it's replaced with this NUS-A, and the core enzyme is able to go ahead and go. So as soon as we have the dissociation of the sigma, we're going to have the NUS-A protein, and this is going to go ahead and allow the promoter to clear, and it's going to accelerate the rate of transcription. Importantly, as we're making our mRNA transcript, the topoisomerase are going to be relieving the stress from supercoiling that we're going to be seeing. Importantly, right, we always have to terminate things. So in this case, we want to be aware of these 2 termination motifs that prokaryotes have. One is row dependent. So essentially what this is that in the mRNA transcript, there's going to be a RUT site. So this is going to be a cis-acting element that can be recognized by the Rho protein. So what this will do is that the Rho proteins will attach, and this will basically kind of function to crawl 5' to 3' in the mRNA transcript. And eventually there's a point where the RNA polymerase is going to stall for a second. And that's enough for a Rho to catch up. And it will just basically, like those magicians that are able to pull a weight cloth, right, from a table, the Rho protein is just going to go ahead and jank out the mRNA transcript from the RNA polymerase. In the case of Rho independent termination, what's actually going to happen instead is that there's going to be this part of the RNA transcript that is able to bind with itself. So it's able to go ahead and bind anti-parallel. And what this will do is that they will form a hairpin structure. These hairpin structures are going to be forming close to where there's just going to be a lot of adenine kind of bonds between the DNA and the RNA transcript. And this will allow it to just go ahead and fall off. And now you have your transcript ready to be expressed. So importantly, we also want to be aware that with DNA, right, we're going to have this major and minor group, and the gene expression that we see is going to depend on how the enzyme interacts with the DNA, right? So it can either interact in the major group or the minor group, right? So here also we just want to think about a few motifs, right? So the first one is going to be this helix turn helix. So essentially what this is just going to be a helix that's going to serve as structural for structure, a turn. Then we're going to have another helix that's going to serve to be the reading helix, and it's going to be able to find or be able to read or find a segment that it understands in the DNA. We can also have this dimeric binding of proteins, right? So in this case, we see these 2 dimers forming, and this allows more recognition, right? Because now we have 2 recognition helixes that are going to be scanning the DNA. So, so now we kind of want to move a little bit into the types of regulations that we're going to see. So what we want to think about is that there can be positive regulation, where either we have an activator that needs an inducer to go ahead and bind, and then this will cause RNA polymerase to have increased transcription, or we can have a ligand that binds to an activator and inhibits it from binding. So in both these cases, we want to consider it to be positive regulation. In the case of negative regulation, what we're going to see is that the end result is going to be the inhibition of transcription. So that can either happen by some repressor binding a co-repressor, and then this is going to stop RNA polymerase, or we can go ahead and have a repressor that has an inducer bound to it. So it's just going to stop it from inhibiting transcription. But when this substrate goes ahead and disassociates, then this repressor can serve and again decrease transcription. So these regulatory elements are going to be expressed basically in front of the promoter, or in the case of activators, a little bit upstream of the promoter. So in the case of an activator, right, this is going to help the RNA polymerase have better binding to the promoter. And in the case of a repressor, this is usually going to be in front of a promoter, so that this can just go ahead and bind and kind of inhibit RNA polymerase from trucking along. So one of the examples that we saw for regulation in prokaryotes was this lac operon. So in this lac operon, what we really want to be aware about is that we have two major players, this CRP protein and a lac repressor protein. Right, so when we have basal levels, right, so what we want to understand with these bacterial systems is that they prefer to have glucose as their preferred carbon source. But when glucose is not available, it's able to go ahead and kick up its transcription of lactose breakdown enzymes, right? So in the case of a normal cell where we have no lactose present, but we have glucose present, we have what we call basal transcription of this lac operon, right? So here we're just making a very small amount of the enzymes necessary to break down lactose. Then what we're going to see is that in the case in which we do have lactose is going to go ahead, and one of the breakdown products of these prokaryotes, right, is this it's going to be this inducer called allolactose. So allolactose will bind to the lac repressor, and this will lead it to not be able to associate with its cis-actin element in the DNA. So if there's no repression there, then RNA polymerase doesn't have to worry about getting over the lac repressor. So, but in this case, again, we still have glucose. So we see there's going to be an increase of transcription from basal to just having a low level of transcription. But when we go ahead and have no glucose present, right, the no glucose present will lead to there being a higher cyclic AMP level. And when there's a high amount of cyclic AMP, these cyclic AMPs will bind to this CRP protein, and they will serve as a co-activator for CRP protein, which itself is an activator. So it will bind to a cis element in the DNA, and it will promote greater transcription of the lac operon, right? So here we see a much higher induction, right? And we see that this is almost 5,000 times more product that would be made from this. So here are some additional notes about the CRP protein. And lastly, right here for the lac operon, we just want to be aware of these three important sites, the O1, the O2, and the O3, right? So all these are going to be bound by the lac repressor protein. And importantly, right, if we go ahead and destroy O1, then this is going to go ahead and inhibit the regulation of the lac promoter. So we would just expect that we would not be able to undergo we would not be able to regulate the expression of these lac enzymes, lactose breakdown enzymes, if we have some sort of mutation that destroys this O1 part. But with that, guys, I'll leave it to I pass on to the next person. And yeah, so
can we ask another question before?
Absolutely.
What in the lecture she mentioned a scenario question that it was when there's no glucose and there's also no lactose, what would be the level of transcription?
Yes, so we can go back for a second, right? So when we have no glucose and no lactose, you said, right?
Yes, someone actually just answered it in the chat. So.
Oh, perfect. In that case, guys, I'll pass it over to the next Desai, and I just want to wish you guys the best of luck with your exam preparation. And thank you so much, guys, for coming today. And yeah, I'll go ahead and pass it to the next person.
Awesome, thanks, man. Let me share my screen real quick, and we'll get started for lectures 44 through 46. So 44 through 46, we're talking about eukaryotic transcription and gene regulation. So specifically, we talked about the actual process, but here we're talking about the logistics of it and how we can make use of it for gene regulation. So primary transcripts basically just the first product of transcription, right? So it's not all the pieces of the primary transcript that are going to be included in your mature mRNA, right? So for differences between exons and introns, the average length of an exon is going to be about 170 base pairs, but for introns, it's going to be mostly 5,000 base pairs, right? And then 10% are going to actually measure up to about 11,000 base pairs or over that even, right? So they're going to be most of your primary transcript, okay? And as you can see here, we're going to go through this obviously, but just going from your primary transcript to your mature mRNA and then to your protein, that's going to be a very important process. So chromatin structure, so chromatin is going to be either an open or a closed state, right? And that's just going to depend on like if it's going to be ready for replication and it's going to be ready to be used, those genes, or we're going to have it in a closed phase where we don't want to use those DNA molecules that are in there. So not all genes are going to need to be transcribed all the time, right? So that's why we have the open chromatin that's going to be 10 nanometer fibers, and that's going to be euchromatin that's available for transcription. And then we have a closed chromatin at 30 nanometer fibers. Those are going to be heterochromatin, closed to transcription. So by closed, I just mean it's going to be inaccessible to most of our activators or remodeling complexes. So if you remember SWI/SNFs, that's what I'm talking about here. And then it's going to be wrapped around those histone proteins. So if you remember those histones 2A, H2B, H3, H4, and the stabilizing histone H1. And histones are going to be covalently modified. So that's not going to affect our actual DNA sequence, but it could still affect our gene expression. And that's where we see epigenetic differences between generations. So for activation, we have to make it into that open form, that euchromatin. So you take your chromatin and you're going to use these transcription factors, these TFs. They're going to recruit RNA polymerase II to the promoter. And we have a bunch of other factors coming into effect here that are helping us make that heterochromatin into euchromatin. So we have that mediator, that polymerase II, and all the trans factors from the pre-initiation complex basically that are required to activate the transcription from DNA to RNA, right? So changing it itself is going to be through acetylation and those remodeling complexes. So as you can see here, these are epigenetic changes. The N-terminus of H3 and N-terminus of H4 can be modified, and then we have both the N-terminus and C-terminus of H2A and H2B that can be modified. So this is what I'm talking about. You have your DNA strand right here, and you have the pre-initiation complex with all those factor proteins, the transcription factor proteins, and you have your RNA polymerase II and your mediator, and then you're going on to go through transcription. And then heterochromatin, just putting those acetyl groups onto it to make it into euchromatin. So histone modification, we have these histone acetyltransferases, or our HATs, right? They transfer that acetyl group onto the lysine of the histone tail. So if you think back about how our histones are connected to our DNAs, right? So we have these electrostatic interactions. Basically, the negatively charged phosphate group that's on the DNA backbone is connected to the positively charged lysine that's on the histones. And that's what we're doing here. We're basically getting rid of that connection by acetylating that lysine residue to become neutral instead of positive. And that gets rid of those electrostatic interactions and lets our DNA become kind of more spread out. And then the opposite of that is going to be using the histone deacetylases, HDACs, and those are going to remove the acetyl groups from the histone. And that's just the opposite action of the HATs. And then another weird one is going to be our histone methyltransferases, or HMTs. Those are going to transfer a methyl group onto the lysine or arginine of a histone tail. So that could actually add stability, and a lot of the times in our eukaryotic cells, we're going to see that the older DNA strands are going to have those methyl groups, while the newly synthesized one are going to be unmethylated. That's kind of how we know when we go into DNA repair. That's a big factor as to what we end up fixing. So the unmethylated part would be more likely to be fixed a lot of the times. And methylation also has a whole bunch of different functions. It could stabilize, it could block, it could activate. So, but specifically for the histones, it's going to add stability. And we also have other modifications we could do. So phosphorylation ubiquitination if you want to degrade that histone. So understanding how we name them and what we mean by these different histone codes, we have an example here. So H3K4Me2, that's going to be histone 3, lysine 4, and it's going to be methylated twice. And for our H2BK15AC, that's going to be our histone H2B, lysine 15, and it's going to be acetylated. A lot of the times you could just be given, like we're going to see later, a question where this goes to an unacetylated version, basically, or a deacetylated version. So our hats, once they're recruited by a pioneering transcription factor, they can acetylate multiple histones. So they're going to work, like we said, in conjunction with the Swiss-Nifs or the remodeling complex, and you're going to need both of these in order to have that open form euchromatin. And the three functions you need to know for the remodeling complexes are going to be unwrapping the DNA from the nucleosome, repositioning, and removing the nucleosome. So those are very important for you guys to know. And the areas of DNA that are going to be without the nucleosomes are going to be ready for transcription. So once you've done that, you're ready to go. And just another picture just to help you out with this. So hats acetylating, and then HDACs are going to deacetylate and then make that heterochromatin, and then here we have the euchromatin. And then same thing is going to happen here. We're just also adding those transcription factors and those remodeling complexes to make it a lot easier to do transcription. So methylation, a big example that Dr. Duma gave in lecture was CpG islands, right? So as you can see here, just from this picture, these CpG islands that are unmethylated are going to lead to the normal gene expression of this gene that comes after them. But once you methylate them, they're actually going to repress that gene expression. And the hypermethylation of the cytosines specifically in CG dense regions near the promoters, that's what's going to lead to that silencing of the gene that's right here. So CG dense regions, we usually call them CpG islands. And that's why we're going to have these islands right here, right before that promoter, and just methylating them or unmethylating them, that's going to cause a lot of gene regulation. Cis-acting elements, like Andreas said, they're just part of that DNA sequence. So they could be promoter proximal elements that transcription factors are going to bind to. And those kind of start at the +1 region or site, and that's going to be included in the promoter region. And then also enhancers are going to be upstream of that promoter to help with gene regulation, basically. Those are going to have binding sites for transcription factors. As you can see here, we have our cofactor that's binding to our transcription factor, that's binding to our cis-acting element, in this case, an enhancer, and that's going to lead to our actual RNA polymerase II attaching to our DNA and making that RNA at the promoter region. So for our transcription factors, they're going to have basically that's going to be referring to a broad range of DNA binding proteins. And they're going to include two specific regions that you need to know. So we're going to have the DBD or DNA binding domain with the recognition helix for the cis-acting element or the DNA itself, and then the activation domain where you have all your co-activators, your cofactors that are going to bind to it to make sure that it's actually recognizing the DNA and it's acting on it. So our classic zinc finger motifs that we see here, those are going to have two beta sheets and one alpha helix. And that's going to be two cysteines and two histidine residues that are coordinating the zinc molecule right here. And then usually we have a phenylalanine or a tyrosine and a leucine that form basically a strut to position this recognition helix, right? So this is our DNA and this is the recognition helix that's acting on it. And multiple zinc fingers can actually work together in order to result in this motif being present. And there's a lot of these motifs, a lot of different ones. I would recommend just going over the slides. You could, if you have time, if this weekend you think you're ready, if you have time, you could just look up some pictures of the different motifs. But I think the lectures also do a great job of letting you know how different they are and what the differences are. And I would just be able to distinguish between them if you're given a question on that. So here's a quick question for you guys. True or false, cis-acting elements are diffusible, so they can function at multiple sites in the genome?
you done?
Does anyone want to yell it out real quick? What they think?
False.
False, beautiful. Okay. Yeah, it's going to be false. So transacting elements or transcription factors, those are going to be what are going to be diffusible. The cis-acting elements, like we mentioned, they stay on that genome. They're part of the DNA itself. So lecture 45, we're just continuing on with our eukaryotic transcription and gene regulation. So we have these trans factors. Again, you have DNA binding domain, the activating domain, and this connecting domain that connects both domains to each other. So the multiple steps that are required to fully activate transcription. We have first the binding of the activators. So the transcription factor is basically just binding to that promoter proximal region or the enhancer, and then recruitment basically of other transcription factors that are going to come in at different parts of the DNA or come in as activating factors at that activating domain. And we also have our acetyltransferases and our Swiss SNFs. Additionally, we have our mediator, just big complex that tries to help us out with transcription, and then the pre-initiation complex on the DNA itself. And it's important to note that the activation domain of a trans factor is the place where these co-activators on other pre-initiation complexes can bind. So a good example of transcription factors is going to be our glucocorticoid receptor, and that's going to act as an activating transcription factor. And it's going to have this nuclear receptor motif to it. And basically what it does is the glucocorticoid receptor is going to bind to this hormone response element at the promoter proximal regions or the enhancers. So those would be our cis-acting elements, and the GR would be our transcription factor. And once it's bound to it, it can recruit a co-activator to bind to its activation domain. So like we said, it just needs something to activate it real quick. And the co-activator and the GR themselves, they basically recruit other proteins. So it's a signaling cascade, basically. And then once that happens, you have the pre-initiation complex bind, and the transcription of target genes can occur. So the pre-initiation complex, pretty huge one, really important to know. The mediator is going to be just this large protein complex that can make those protein-protein contacts with the transcription factor activation domain. So basically just what connects the transcription factor with the activation domains and the proteins, and it's going to largely interact with the pre-initiation complex. So the rest of it is going to have a whole bunch of different domains, right? And a good tip for this is to make a flow chart to remember what order all the TFs bind, the transcription factors, and just to know also their different functions and how they help us in the process of transcription. So TF2D is going to bind at the promoter, and that's basically just binding at the TATA boxes when available. So this is why it has this TBB kind of name to it. So TBP is just TATA binding protein, and it's going to also bind at the minor groups specifically. And now we have these staffs. So basically just those TATA binding protein associated factors that are going to be associated with this TATA binding protein. And they're going to recognize other sequences and act on them. Right after that, we're going to have our TF2B bind to the DNA and their TBP as well. And then once our RNA polymerase II joins the complex at TF2B, now we're ready to add TF2E and TF2H. And I would say TF2H has two really important functions. So TF2H, H for helicase. So it's going to have a DNA helicase activity that's going to unwind that DNA and get ready for transcription. And then we have our kinase activity to phosphorylate the RNA polymerase II to initiate transcription. So this is a great example that Dr. Duma uses in her lectures. So for gene activation, we're going to have in yeast, we're going to have this HO gene or HO gene, if you're into that. So yeast is a eukaryote, right? And that's a great example of our remodeling complexes, Swiss SNFs, as you can see here. So the first thing that's going to happen is going to be those pioneering transcription factors, SWI5 actually in this case, that's going to bind to that upstream enhancer or that cis-acting element. Once that is bound, basically it's going to recruit our remodeling complexes, our Swiss SNFs, and then this GCN5 complex, basically just a histone acetyltransferase, is going to enter to decondense that heterochromatin and make it into euchromatin. And then we have this SBF activator that binds at the 5' promoter proximal region. Again, another cis-acting element, and that's going to recruit our big, large complex, our mediator, basically. And then after that, our RNA polymerase II is assembled at the region free of nucleosomes. Remember, we can't have them bind to regions full of nucleosomes. And then finally, transcription can finally start, right? And then the activation or the activator binding is going to be sequence specific. So just one little single nucleotide polymorphism, or SNP, can inhibit the binding of an activator enough to change your entire phenotype. So it's going to be a very important process, and that's why we come in with DNA repair and fix any single nucleotide that's not the way it should be, because it's going to affect you the whole way through. So for repression, we have these receptors that are going to do the exact opposite as our activators. They're going to inhibit transcription, right? And they compete with the activators for binding to a regulatory site. They can actually bind to the activator itself to prevent the interaction with the mediator, or they can alter the assembly of that pre-initiation complex we just talked about. And they can also provide a docking site for HDAC, which, if you guys remember, that's just what deacetylates those histones and turns them back into heterochromatin. And then a repressor has this DNA binding domain to bind a negative regulatory element. So what that means is basically you have this repressor, it comes in at this DNA binding domain, it comes in at the cis-acting element that binds, that's a receptor specifically for that repressor. And then once it does that, it's going to block this general process of transcription factors and RNA polymerase II binding to the DNA and basically targeting that gene for transcription. So a quick question for you guys. Following transformation occurs, all of the following are likely to be true except. And someone can just yell it out once you guys got it.
Is it A?
Yeah, it's beautiful. Yeah, it's going to be A. So your heterochromatin is not going to transform to euchromatin, right? Because we have this acetylated version, and then we go to the deacetylated version, and that just means we went from euchromatin to heterochromatin. And then what the rest of it is going to be true because you're converting this 10 nanometer to 30 nanometers. So just going from our euchromatin to heterochromatin. And then the chromatin is going to become less available for transcription. As we know, heterochromatin is condensed. And then a repressor domain is going to be nearby. So that just means that this repressor basically affected our DNA in a way where it doesn't get transcribed anymore. Perfect. So for gene control, we have a bunch of different ways. And you kind of get to those ways just by knowing what we've just talked about. But the condensation to heterochromatin is going to silence those genes. And a good way to picture this is if you imagine just kind of in a lab setting, you have the heterochromatin that's staining a lot darker than a euchromatin, basically. And that's going to tell you that it's a lot more dense and it's a lot harder to get to the actual DNA in there. And then histone methylation is going to stabilize the DNA in the condensed form. So it's going to make it harder to go through transcription. And then the level of transcription of a gene itself is going to depend on the occupation of regulatory elements of the DNA sequence. So basically, just those cis-acting elements and how they're put together on our actual DNA. And this is kind of getting more at the idea of combinatorial control. And it's just telling you that humans are going to have far fewer transcription factors than genes. So we have to mix and match those trans-acting elements. And we usually have more than 6 regulatory elements. So it's less about those transcription factors, and it's more about the regulation of them and where you put the promoters, the enhancers, and how they interact with the transcription factors. And then finally, each cell is going to react differently to different transcription factors. So they're going to have different receptors, different secondary messenger systems, and that's how we have our differentiation between different tissues and different organs. So for lecture 46, we talked about post-transcriptional RNA processing. So what happens after you go through transcription, right? So RNA polymerase II basically took your DNA and made it into this primary transcript. Now, it does not know the difference between exons and introns. It's just turning your DNA into RNA, right? So just giving you this whole big strand of introns and exons, but it doesn't really know which to keep and which to get rid of, right? So once we do that, we're going to go through a whole bunch of post-transcriptional modifications to get to our final mature mRNA. And I just like to point out real quick that this process is exactly the way it is for shorter transcripts. But once you get to longer transcripts, this RNA splicing that you see here is going to happen a little bit early on. It's going to be occurring all throughout the process, and rather than just at the very last step. But yeah, basically you're going to export the mRNA itself to the cytoplasm for translation and turn it into a protein. And yeah, those modifications, we're going to go through those. But first, before we can do anything, we have to work on the C-terminal domain, or CTD, of the RNA polymerase II. So this just has to do with our termination steps and then how we get to our post-translation or post-transcriptional modifications, right? So we phosphorylate the serines that are going to be on that CTD, and that's going to be necessary for a whole bunch of different things, right? That is going to help us with promoter escape, so just getting rid of that promoter that got the process started in the first place, and then it's going to act as a docking site for a whole bunch of enzymes. So we have our capping enzyme, so it's the 5' capping enzyme, and then our cap binding complex, our termination complexes, and also our RNA splicing factors. And for the capping enzyme specifically, that's going to assemble our 5' cap and provide basically protection from exonucleases, and it's going to be really important to initiate translation. So a closer look here at how we terminate transcription. So we're going to have a DNA sequence at the very end where we want to stop, basically. That's going to be TTA-TTT, right? That's going to correspond to an mRNA or an RNA sequence that's going to be AAU-AAA, right? And that's going to be your signal for termination that's going to be located in that newly formed RNA. Once that is basically encountered by our RNA polymerase II, it's going to go a little bit past it, and then it's going to terminate. So our CTD-bound termination factors that, remember, we needed that phosphorylation of CTD to put them on, those are going to recognize the cleavage sequence, basically, that's right here, and it's going to bind to the RNA polymerase II, and it's basically just telling it, hey, like, we got to go, like, we're done here. And the cleavage of the completed primary transcript is going to happen, and then the RNA polymerase is going to dissociate after those poly-A polymerases are bound. So the poly-A polymerases, we're just adding that poly-A tail that we know about at the 3' end. And then once that is bound, our RNA polymerase II is going to dissociate. For our introns, so we have these splice junctions that are going to be linking our 5' exon right here and our 3' exon right here. And those are going to be basically just to get rid or excise those extra introns that are in our primary transcript and make our mature mRNA. So the GU-AG rule, that's just basically what we're going to use to splice those introns out. The 5' end of an intron is going to have a GU in it, and then the 3' is going to have an AG. And within the intron itself, we're going to see this branch A or branch point A point that's going to be usually closer to our 3' exon or our 3' intron side than it is to the other side. And that area is going to be kind of followed by a pyrimidine-rich region. And as you can see, it's between those, between the actual branch point and then the 3' splice site of the intron exon. So the splicing mechanisms, we're going to use these SNRNPs or SNRPs. So we have U1, U2, U4, U6, and U5. And our U1 is going to be our splice site itself, right? So as you can see here at the GU, or our 5' region, basically, we have our U1 or our splice site. And then we're going to have our U2 at the branch A site. So our U2 is just going to be right here, and our U1 is going to be right here. And then after that, we're going to have our U5, U4, and U6 that are going to then bind. And Once they're bound, we're going to create this lariat looking structure right here. And that's going to be the inactive spliceosome. So we're still not in the active form. That's really important to know, that once our U1 and U4 pop off, basically, so once you go through this step and our U1 and U4 exit, then you're going to have the active form of the spliceosome, which is just going to be your U2 at the branch point A, and then your U5 and U6. So the first reaction that happens after we have the active spliceosome is going to be between the branch site and the 5' site. So the intron is going to be produced in this lariat structure. So this yellow part here, that's going to be your intron, and then the green is going to be your exons. Once that happens, we get this second reaction between the 5' and the 3' sites, basically just the exons that are left. And we want to get rid of this whole active spliceosome complex and also our intron. So that's what's going to happen. We're going to see that release happen, and then we get our 5' to 3' mature mRNA or exon only RNA. And then for alternative splicing, so this is going to be our mechanism, basically just for variation. So how we take one DNA, or let's say even primary transcript of mRNA, and we turn it into just a whole bunch of different functional proteins for different organs. So your liver, your brain, they could use the same actual transcript, but they can produce differently, or proteins that do different functions, different things. And 60% of all mammalian primary transcripts are going to undergo alternative splicing. So the green boxes here, those are going to be your exons. And the yellow here, as we saw in the last example, those are going to be your introns. And what happens here is just we take these exons, we put them in different orders, we make these two versions, basically these two copies that could be different, and we use them for different things. And there's only really this one example we know that we retain that intron, or intron retention. We would just keep the one intron in there. I would go through these, I would know them, and they're not that hard because they kind of make sense if you go through them. And I would know them for the exam. They're kind of a freebie, just in case. So our last thing for this lecture here is hemophilia. So the gene for clotting factor 9 is going to be X-linked, and it's going to be established as a hemophilia-associated gene. So basically what happens here is this splicing defect is actually going to result from an error in the selection of a 3' splice site. So a normal individual would have this, this sequence right here with the splice site being right here, right? But then a royal hemophiliac would have the splice site be right here. So it's going to alter the way the way the actual frame shift or frame is read, basically. So the open reading frame is going to be read wrong, and that's what's going to give you that loss of that factor 9 and basically result in hemophilia. So one frame shift mutation is going to result in this loss of the entire activity of the factor. And then with that, we can move on to lecture 47. And yeah, who is next? I think it's either Kishan. You there?
Yeah, I'm next.
Yeah, beautiful.
All right.
Beautiful, man. Take it away.
Okay. Are you guys able to see this? If you guys could put in the chat box. All right, sweet. Okay, so today I'll be talking about translation. So that's basically going to be like the last part. If you think about just like the central dogma in general, from translation, we're basically making proteins. So some key things for translation in terms of like the mRNA, there's going to be a series of 3 bases. And so terminology is like really important for this exam. So you might just get like a couple easy questions and just free points just from knowing the terminology, like knowing what a codon is. So a codon is typically going to be associated with mRNA, and it's going to correlate for an amino acid. And so typically these codons are going to be in something called a coding sequence. And you guys should know what your start codon is and like the 3 stop codons. So the start codon is going to be AUG. And then the stop codon is going to be UAA, UAG, and UGA. What I told my students, if you guys also want to use this, is that like a good way to memorize the stop codons is like you are annoying, you are going, and you go away. It's just something that works and worked for me. So if you guys want to just know that, but because you guys have to know like the 3 stop codons. But in general, there's like 3 aspects of the genetic code that I want you guys to kind of focus on in terms of translation. And that's one of them being that it's going to be non-overlapping. So what that means is like, if you look at like a coding sequence right here, it's going to be non-overlapping, as in you're going to have like three distinct like groups essentially. Like you're going to have three, you'll have like codons essentially. They're grouped in codons, or three bases. And so they're not overlapping because, like, for example, if this first group of three and the second group of three were overlapping, and you had like A, C, and G, like if this was a group right here, that would be overlapping, right? And the thing is, if this was to be true, like this would actually code for a different amino acid based on like that codon chart. And so that would honestly just like mess up your protein, because you would just have a different amino acid than the one you need. And then in terms of the genetic code, it is universal. So it's going to be like primarily seen in like obviously the prokaryotes and eukaryotes both. And genetic code also is considered degenerate. So what that means is like we'll have 64 codons, right? But we only have 20 amino acids. So what that basically means is like we're going to have multiple codons for amino acids. Typically, most of your amino acids will be like we'll have multiple codons essentially, and like we'll see that through the chart. So next, mutations are really important. It's just like a freebie, I would say, in terms of mutations. And there's like two types basically that you guys should know, and it's the single base substitutions and frame shift. So single base, like it's kind of in the name, you're going to basically have like a substitution of one of the bases. So like, for example, you have nonsense mutation, and a good way to think about nonsense is it's like it's nonsense to stop right now, or like to stop like essentially translation. And so what that does is you're going from basically any type of codon to a stop codon. And here you can see like the single base substitution is going from G to U, right? So you're going from a codon to, and you're introducing a stop codon, basically shunting like the process. So it's like a nonsense to stop, like you want to keep going in order to like, you know, make a polypeptide chain. For missense mutation, you're basically, think about it as you're going from one amino acid to the other through a single base pair change. So you're going from like G to U in this one as well. And you're changing from one amino acid to the other. And silent mutation, it's like typically not really considered a mutation, but like it technically still is, because you are like altering like the base sequences and stuff like that. But you're going from A to U in this like example right here. But basically the main thing is you're keeping the same like amino acid. Like both of these codons encode for glycine, so you're not really like switching anything up, and it's not going to have like an impact on the protein in general, but it's still considered like a mutation, hence like silent, because there isn't really an effect. And then in terms of like frame shift, so frame shift, there's going to be insertion and deletion. Those are like the main ones, and it's kind of just in the name. Like you'll have either an addition of like 1 to 2 bases like randomly within the mRNA, and that'll obviously like rearrange the groupings, right? Because we want them to be grouped in like pairs of 3. Or deletion as well, which is just removing 1 to 2 bases. To be honest, I like never really knew what like an in-frame deletion or insertion was, like until I watched like Dr. Duma's lectures. But basically like an in-frame deletion or insertion is like 3 bases at a time. So it's not going to be like as like volatile, or it's not going to like really affect you that much. It's still considered a mutation because you are removing like 3 at a time, but it's better to remove like, you know, those groups 3 at a time rather than just like 1 or 2 bases, which will lead to like obviously more of an impact. So reading the chart, this is like super important. I just want to take like a few minutes just to quickly just go over how to read this chart. So if they give you something like, I don't know, like ACA, you'll have like your first letter in your codon. So you'll have A. And so you're looking here on the vertical axis. So you have A right here. And then your C, you're going to be looking for your second letter is going to be on this like horizontal axis. So you have your C here. And so ACA, you see like where they kind of converge and like meet together. And it's going to be this square right here. And then you just look for ACA. All right, I see it right here. So it's going to be coding for threonine. So that's how you should like kind of read the amino acid, like this chart of codons. And this is going to be your start codon. And then these are going to be your 3 stop codons, which you guys are responsible for knowing. So just a question for you guys. If you have a codon and you're going from here to here, like what type of mutation is it? And like you can use this as a reference. And you can just put the answer in the chat box. Yeah. All right. So the answer is E. So good job to those that got E. And so the reason why it's E is if you look at like the 2, right? There's only a difference in 1 base. So automatically I'm already thinking like it's going to be either nonsense, silent, or missense, because those are examples of single base mutations, and you have a single change like from A to G. And so like if you look at it, so we're looking at like CAU, right? So C and then A and then U. So that's going to be coding for histidine. And then CGU is going to be coding for arginine. So you're going from histidine to arginine. So anytime you like, what do you call it, switch between one amino acid to the other, it's going to be known as the missense mutation. So that's why E is correct. So now in terms of experiments, they like asking questions about experiments. And I know there was already presented like earlier, there's a bunch of experiments going on in like, I guess this exam in general. So they could ask you about like the genetic code and how it was cracked. So it was actually cracked by someone named Marshall Nirenberg. So like just essentially what he did was he used like a bunch of like poly-U, a poly-U's, which is just like a bunch of U's, a bunch of bases that had U's in it. And he had used like the cell-free extract. So if you guys remember the cell-free extract from the, I believe like the first lecture, where it basically has like, it's basically like a lyse cell, but it has like all the proteins in it and enzymes in it. And then he also used ATP or GTP as the source of energy. And so like there's like 20 of these tubes, and each of these tubes, so there's like 20 tubes for like 20 amino acids. And so what he did was he tried to see like which one would typically like react given like these like substrates, right? And he saw that like from the phenylalanine tube, he got something called polyphenylalanine. So he knew immediately like that was due to using the poly-U's. So he knew that like the phenylalanine was like the codon for phenylalanine was UUU. And then he did the same thing with all the other amino acids. And this is like how he started to find out like what the specific codons were for these amino acids. So like for polylysine, he found out that poly-A encodes for it. So it's going to be like AAA. And then proline is going to be CCC. And like you can double check this as well in the chart. So UUU is going to be like phenylalanine. And then what was the last one? It was like AAA. And you can see like AAA, which is right here, is going to be for lysine. So that's like exactly like, that's kind of like what he did. But yeah, it's pretty cool. So in terms of tRNA, so tRNA, you should know, like you should be a little familiar with like the structure. So it's going to have like a clover leaf structure. And just like, I guess like the answer to this question here, this was a question that I had for like my students, but tRNA is typically going to be associated with anticodons, and then your mRNA is going to have the codons. And so like the structure for tRNA, you're going to have an acceptor arm, your D arm and loop, and then the T5C arm and loop, and then the anticodon loop. But like this acceptor arm, this is where your amino acid is going to be. And that amino acid has to do with obviously the anticodon that's like a part of this. And like you can also see that like there's what do you call it? There's like inter-strand base pairing between like the 5' and 3' ends. Like there's within like the tRNA molecule, there's like that base pairing. But like the more important point is like the concept of a wobble base. So what you like in terms of like anticodons and codons, they're going to be base pairing in anti-parallel like directions. So what that means is like you have 5' to 3' for anticodon. And just note, like this is just a picture. Your 5' could be on this end and your 3' could be in this end. But just in this picture, you're going from 5' to 3' for your anticodon. And then your codon is going to be going from 5' to 3', but in the opposite direction. So this one's going left to right, and this one's going to go right to left. And you have to know like which position the wobble base is at. And that's going to be the 3rd position at the codon. So this is the wobble base. And the way you can figure out like the numbering for that is if you number from 5' to 3', it's going to be 1, 2, 3. So this is like 3rd base. And that 3rd base is going to be base pairing with the 1st base of the anticodon. So this is 1, 2, 3, because you're going from 5' to 3'. So this 3rd base is known as the wobble base, and it's going to be in the codon. And what it does is it allows for more degeneracy of like the genetic code. So you might be like, you know, like, what does that even mean? And so what that means is like, anytime you have a wobble base in here, you can actually bind, like you can have a, like this base could be like any type of base depending on what this like base is in the anticodon. So like, for example, if this was a U, this could be an A, or it could also be a G. So these are like all the different combinations in which you can have, like these are like non-Watson-Crick base pairing. So they go against like those like Watson-Crick base pairing rules. And you also have something called like inosine. Inosine, you can see like literally can bind to A, U, or C. And so like this also helps with like having fewer tRNA molecules, because like obviously we have 64 codons, but we don't have like 64 tRNAs, right? And so we're not going to have like 64 tRNAs like carrying those anticodons or like 64 codons. So like if you have an anticodon right here, which is like UAI, for example, you can see that this I can literally cover like 3 different types of codons, because this is like the wobble base right here, and you can see like it binds to either the U, C, or A, this inosine. So that just allows for fewer tRNA molecules in general. So that's like the whole like concept of like the wobble base essentially. So in summary, like in terms of translation, just like an overview, I guess, is that you're going to have an activation, initiation, elongation, and termination. So activation, we want to first charge our tRNA molecules, and then they're going to basically go to the ribosome. And then we're going to have the initiation. Basically what happens is you'll have like a 16S rRNA binding to like the Shine-Delgarno sequence of your mRNA. And then you also have like your start codon, which is your AUG, bind to like the P site of, and that AUG is going to be in your mRNA because that's a codon. And then like elongation is kind of within the name. Like the whole purpose of this is that you're building a polypeptide chain, right? And then termination is going to be like, we're basically done. And that's typically associated with a stop codon, right? So those 3 stop codons that you guys are responsible for knowing. And there's going to be like release factors and stuff like that, but I'll get into the details for that. And then we'll obviously have post-translational modifications as well. So I kind of use like the diagrams from the lecture because they kind of really helped in terms of understanding like the translation process. So this is going to be the process of charging. So really quickly, like one of the names of the enzymes that you guys are responsible for knowing is like the aminoacyl tRNA synthetase. And so what happens is you'll have your amino acid right here, right? And this is your ATP. So remember, Nuremberg used ATP and GTP. So this is kind of where that comes into play. So you have your amino acid, this is your valine, and it's going to be bound to like this enzyme, and you also have a separate binding pocket for your ATP. What's going to happen is your pyrophosphate is going to be kicked out, and you're going to have a bond between AMP and your valine. And so now like there's an empty pocket right here, and this is going to be for like any free floating tRNAs. So we're going to actually try to find from the environment like a tRNA that has specifically the anticodons for valine, right? Because valine is like the amino acid that we have. And so we're trying to look for like the tRNA with these specific anticodons. And so once we find that, it's going to bind to the pocket. So it's going to have like the structure right here. This AMP is going to be released because we want this amino acid to be bound to the tRNA. The reason why we want the amino acid bound to the tRNA right here is like once it leaves, it's going to go straight to the ribosome, and then it's going to find the corresponding codon that it can bind to on the mRNA, like on the ribosome. So that's the whole process of like charging essentially. So in terms of ribosomes, just really quickly, in terms of like the bacterial ribosome and eukaryotic ribosome, you guys are responsible for knowing like the bacterial one is 70S. The big subunit is going to be 50S, the small one's going to be 30. And then for eukaryotic, it's going to be 60S for the bigger one, and then 40S for the smaller one. And just remember, these are not additive. So it's not like, 50 plus 30 is going to be, that's going to be 80. But obviously the ribosome is going to be 70S. So just don't think of them as like additive, but just know like which one is the bigger one and which one is the smaller one for both. And a good thing for like in terms of all the processes like for this exam, what really helped me was having a comparison, like a kind of like a T-chart of prokaryotes and eukaryotes. And I did that for like each of the processes, like for transcription, replication, translation, like all of those, because they'll like to ask like differences in like the two. And I'll like go over those as well when I go through translation. So in terms of initiation now, so we already charged the molecule, so now we're ready for initiation. So obviously, hence, like with initiation, we have something called initiation factors. So initiation factors, think of them as like blocking the subunits for the ribosome. So the three sites that you have to know is EPA. I just like to remember as like Environmental Protection Agency, but it's just like EP and A. E has to do with exit, P has to do with like the peptidal tRNA, and then A is like your aminoacyl tRNA. So what you have is like your factors right here, and they're going to bind to each of the sites. So your IF1 is going to bind to the A site, your IF3 is going to bind to the E site. And the reason why we do that is because we don't want any like tRNAs to immediately bind. Our like whole purpose of this is we want to load the mRNA first and then put the tRNA. And this is in prokaryotes. Just keep that in mind because it's going to be the opposite for eukaryotes. So we have these factors bind. Once they bind, they make sure that any free floating tRNAs don't bind, or any of like the 50S ribosome doesn't come in. The larger subunit doesn't come in like bind to like the smaller subunit. And so what happens is you have your 16S rRNA. So this is like the rRNA that's a part of your 30S or your small like subunit. And this is going to be your Shine-Delgarno sequence that it's going to bind to. And that's going to be on the mRNA molecule that we're trying to load onto the ribosome. And your starting codon, always like know that like your starting codon, which is like your AUG, is going to be lined up in like that P site. And so essentially something called IF2 is going to bind with GTP. So this is like where the GTP comes in. You remember, like we use ATP for charging. We're going to use GTP for like the initiation kind of step, just like as like Nuremberg used. And so like GTP essentially is going to be bounded to IF2, and that's going to recruit this tRNA. One important thing that you guys should know, that I feel like you guys will see on your exam, that could be like a trick question, is that like the first kind of what do you call it, amino acid that's going to be like made from like the prokaryotes in that chain is going to be methionine, but it's actually F-methionine or formal methionine. So it's going to be actually F-met in prokaryotes, and that's going to be associated with that AUG. Typically we associate AUG with like methionine, right? Like based on like chart, but like in prokaryotes, it's going to be F-met. That's like the key thing. That's going to be the first one. So that's really important as like a distinction between prokaryotes and eukaryotes. But anyways, once you have your tRNA, it's going to come and bind to that P site. And you can see like this is the anticodons and these are the codons, and they're going to be bound together, right? So these are going to base pair pretty well. And then, so after that, we basically have all these, like, once they're bound, we want to get rid of all these like factors. These are just blocking, like we want entry, right? We want these like free floating tRNAs that are out here to bind to the A site. And what I like to tell my students is like, when you have an A site, I like to think of it as like the acceptor site, as in your tRNA molecules are going to come into that acceptor site, that A site, in order to make like that peptide bond between the A site and the P site, which I'll go over. But basically, once your factors, your initiation factors go away, like they're going to be released because your GTP is going to be hydrolyzed. And so once that happens, your 50S is going to come in, and this is like your whole like initiation complex. So I know that was a lot, but if you really like rehearse and like kind of, I guess, like draw it out too, that really helped me in terms of like understanding like how the initiation process like works. And so now in terms of elongation is kind of like where we're going to start building the chain. And so the main thing that you guys got to know is that it's going to be done through EFTU GTP. And so EFTU GTPs, what it's in charge of is it's in charge of like recruiting any other like amino acid. So we already have F-met in your P site, right? So we want another amino acid in your A site with a tRNA. So we're going to look for a random amino acid bound to a tRNA. So this is a charged tRNA, and your EFTU GTP is going to bound to that charged tRNA and recruit it to the A site. So now we got it in the A site, it's going to hydrolyze, and it's going to release and go through all this like whole cycle. But essentially at the very end, like you have another amino acid on your A site, and one, like your starting one, which is your F-met on your P site. And so now like what's going to happen between these two is like what you think is going to happen is when you have two amino acids, you're going to have like a peptide bond. So now you have your first peptide bond, and the ribosome is peptidyl transferase. It's like an enzyme that kind of catalyzes the peptide bond. So you'll have like a nucleophilic attack like this. This like what do you call it? The lone pair on the nitrogen is going to attack this carbonyl, and you're going to have like a whole like peptide bond, which is seen in this picture right here. And this is going to be between the amino acid on the A site and the amino acid on the P site. This is your F-met, and this is just like a random amino acid. And so what happens is like this F-met is like dragged onto the A site. So now we have like both of these combined, but it's going to be on this A site. And the thing is, like, once that happens, these like these tRNAs kind of lean over. So like you'll have like this whole like peptide chain on the A site, but technically it's going to lean over into the P site. And this empty tRNA that originally had F-met is going to lean over to the E site. And the reason why is because this is empty now, like we don't want it. So it's going to be ejected. Like the E site is like the ejection seat. And so now you'll see like they're kind of like leaning towards like the site that they want to go to. So that's what happens. So now in terms of translocase, so obviously like we need to move this mRNA in order for these incoming tRNAs to bind to the rest of these codons, right? So how are we going to move this mRNA? So there's something called EFG GTP translocase. So translocase, like translocation, has to do with movement, right? So this has to do with the movement of mRNA. So we already have it loaded on here, but we need to get through like and get access to all these other codons. So it's going to be moving to the left, right? And so we do this through EFG GTP. And so once it binds, like we're able to basically move it along like to the next, like we're allowed to move the mRNA. So you can see like the AUG moves to the left, and it's like right here now. And now we have access to another like codon that can be bound to like a free floating like what do you call it, tRNA with the anticodon. And so like that's basically what happens is like that translocase is responsible for moving that mRNA. So now in terms of termination, so once we made a long enough like chain at the P site, so typically what happens is once you have like the A site, once like they were leaning, you remember they were like in the A site and we only had two, like a peptide bond, like a single peptide bond, it's going to lean and go into the P site. And now the A site is empty. Every time we want the A site to be empty because now we can have incoming tRNAs come in and like make another bond, and then it'll come into the A site and then go back into the P site. So that's like what happens is we want to constantly like keep this A site empty essentially after like the peptide bond is made. So other incoming tRNAs can come in. But essentially at termination is what you kind of think is we're stopping translation. And that's because we come into contact with these like stop codons. And so release factors, specifically like RF1 and RF2 are going to recognize like certain sequences. I don't really think that's too important to know like which sequences they specifically recognize, but just know that the release factors are going to be released and they're going to bind to the A site. And so after that, like you essentially have like the disintegration of the whole complex. It's going to break down and it's going to be reused again. And you have to go through that whole like initiation complex. Like you have to make the initiation complex again, essentially. So that's basically it for termination. So now in terms of the part 2 of translation, I'm going to mainly go over like the post-translational modifications and stuff like that. But really quickly, I just want to go over like the antibiotics and stuff because they like to ask questions on like clinical applications. So they like asking questions about like experiments, processes and stuff like that, but also they like to see like if you know your inhibitors. And so one of the drugs that are used, it's not actually clinically used because it actually affects us and bacteria. And we only want like, when you're thinking of an inhibitor for translation, you only want it to affect the bacteria because then if it affects us, it's like bad. If we're unable to undergo translation, we can't make new proteins, and then like obviously that's bad for us. So we want to only target bacteria. But puromycin targets both. So puromycin in general is going to be a translation inhibitor. And so if you guys remember from like the translation process that I just went over, you have like the charged tRNA. So what it's going to do is it's going to act like a charged tRNA, and charged tRNAs go into the A site. So we have puromycin in the A site now, right? And it's going to act just like a tRNA. So it's actually going to bind. It's going to bind to like it's going to make a peptide bond essentially. And it's going to basically be added to that new chain, and it's going to stop translation essentially. And then you have like now your 4 clinically used drugs. So you have something called chloramphenicol. So what it'll do is basically block your ability to make a peptide bond. So it blocks that like ability to make a peptide bond. So you can't even like undergo elongation essentially. Erythromycin. So like the E in erythromycin, and I like to think about it as like it blocks like the E, like EFG. So and EFG was that translocase, right? So if erythromycin is used, like the translocation won't occur. And so you can't even move your mRNAs like along the ribosome, which is bad. And then you have your tetracycline. And so the tetracycline actually is going to be blocking your charged tRNA. So I like to think of tetracycline as like T, and then it blocks like the tRNAs. And it's going to block them from entering that A site. We want them to obviously enter the A site in order to make proteins and stuff like that. So that's just another drug that's used in order to obviously inhibit bacteria, which is good for us. And then we have streptomycin. So streptomycin, the S in streptomycin is going to distort the structure. So S for structure and S for streptomycin, it's going to distort the structure of mRNA. So basically, like think of it kind of like a frameshift mutation. Like it's basically going to like disorient the structure. And so our mRNA is going to be read like incorrectly by like the ribosome, or like the prokaryotic one. It will be read incorrectly by the ribosome. So that's what happens like for these like drugs essentially. So RNA interference, I would think about it as regulation. So we have obviously like negative regulatory mechanisms. And so that's kind of what you guys should think of in terms of RNA interference, and interfering kind of has like a negative like connotation, I guess. So you have, and they're going to involve like these small non-coding RNAs. So you have like miRNA and siRNA. So miRNA, I would think about like us, like mammals. And so they're going to be basically what they do is they regulate like whether the mRNA can be translated, and it also regulates the stability. So in us, like in eukaryotes, we don't all like the mRNA that we make is not all going to be translated, right? So we have like select mRNAs that we want translated. And so this also helps in controlling like which ones we want to be translated, and also like turning off like the translation of other like mRNAs. And then siRNA is going to be basically regulating like it in plants mainly, and it's going to undergo like a cleavage, like by an endonuclease. So both of these actually are going to be small non-coding RNAs, and the reason why it's non-coding is because they just don't code for protein, but they rely on something called the RISC complex. So like this is just a picture from your lecture that has like the RISC complex, but just really quickly, you have like the transcription of these like silencing genes. You're gonna have like the formation of like this like hairpin loop structure kind of thing, and it's gonna come and it's gonna be cleaved by something called drosha, which is an endonuclease. And then like this hairpin structure essentially is going to be brought to something called exportin 5. It's kind of within the name, like exportin is going to export. So you're going to basically export like this hairpin structure out of the nucleus and into the cytoplasm. And once it goes into the cytoplasm, it's going to be cut by something called dicer. And like dicer, kind of within the name, you can kind of tell like it's going to be diced. It's going to be like sliced up and it's going to be cleaved. And so you have your RNA, and you have double-stranded RNA now. So we know RNA typically exists in like a single strand, right? And so there's going to be something called RNA helicase that kind of separates it. So now we have like a mature miRNA, and now it can go through two paths. It can go through the miRNA pathway or the siRNA pathway. So if this like miRNA complementary, like has complementary binding, like perfect binding to the target mRNA, then it'll go through this siRNA pathway and it'll just be cleaved. And that's how like this is like all like negative kind of regulatory mechanisms. And then in us, like so that happens in plants, it binds perfectly to the target mRNA. But if it partially binds to the target mRNA, and which occurs in us, if there's partial binding, it's only going to repress like translation. So we obviously have like some repression. And so this is kind of like a whole slide on just like regulation in general. So now eukaryotic translation initiation. So in terms of this, like I'm not going to go through this entire thing because it's very similar to like the prokaryotic one, but the main difference that I want you guys to know, and that I feel like it probably will show up on your exam, is that like for prokaryotes, for initiation, you're going to have first like you're loading your mRNA and then your tRNA. But in here, you're going to load your tRNA and then your mRNA. So it's like a little bit of opposite. And like you guys are responsible for knowing like these factors, but like a good way to tell, like if you're given like something like ELF1 or like ELF3, you can see like the E in here has to do with the eukaryotes. So you know immediately like, okay, this has to do with like eukaryotic initiation, right, for translation. But essentially the factors bind again, they're going to block the sites. And then you have your tRNA. Now you can see a methionine, right? So this is eukaryotes. So the first one's going to be methionine, but if you guys remember in prokaryotes, it's going to be FMET, which is formal methionine. And so once the tRNA comes in and binds to the P site, we have something called ELF4 that's bounded to like this like 5' end of like your mRNA. And so we're essentially, what it does is it helps load it onto like the smaller subunit. So remember for eukaryotes, the smaller subunit is going to be 40S and the bigger one's going to be 60S. So once you load like the mRNA onto here, they obviously like we're trying to find like the part that has like the start codon, right, on the mRNA. So we're going to scan it, right? We're going to keep scanning, and like we see it now. And so now once we found like the AUG in here, which I believe has something to do with like the Kozak sequence, they're going to start binding. And now like once we found the AUG, what we can do is just pop off all these like what do you call it, initiation factors, and essentially the bigger subunit is able to come in and now make like that pre-initiation like complex essentially. So that's like the whole process for eukaryotes for the initiation. So just like this was like pretty cool like when Dr. Duma presented it. So it has to do with like the SARS-CoV-2, which is like the virus that causes COVID-19. So you guys honestly might see a question on this because we are like during the pandemic. So it'd probably be just like wise to just quickly look over this slide, because she honestly might ask it since we are in the pandemic and stuff like that. But anyway, so like what the virus does is it's going to produce a protein, right? It's going to produce an NSP1 protein. So you can see like just through here, like it has like a wide range of like what it's impacting. So you can see like it has something like a linker, right? It has like a flexible linker that's going to block this like site in the ribosome. So what I want you guys to think of when you look at the NSP1 is it's going to be a translation initiation inhibitor. And the reason why is it's going to compete with the mRNA. So typically the mRNA would be on here, right? In translation, like the mRNA is going to be loaded onto the ribosome site, the binding site. But now we have like the NSP here, and it's like tampering with this binding site, which is bad. So that's basically going to reduce the amount of these ribosomes that can actually undergo translation. And the thing is, when we're comparing our translation, our cellular translation, with the viral translation is actually favored, unfortunately. And so if they do take over with this NSP protein, it's favored. And the reason why is because they have like certain stronger like and more efficient signals for like the initiation of translation, which allows like for more of their ribosomes, their viral ribosomes, and like their viral kind of proteins to be made essentially over ours, which is unfortunate. So now in terms of just other inhibitors, we have ricin. So ricin's like one of those like most deadly natural compounds on the planet. It's like crazy how like less than like 2 milligrams of it can like have like a 50% chance of killing someone. That's kind of insane. But basically it can be ingested or inhaled. But the main point is like what I put in red. You guys should know like how ricin is, how like, I guess it's like how it's performed, like what it does essentially, its function. And so it's a protein, right? And so it enters, and when it enters, it's going to be proteolyzed. It's going to be broken down by things called proteinases. And so it's going to release something called RTA. And RTA actually just messes with the binding site. It's going to depurinate the binding site of which that translocase factor can bind to. So think about it. If you like destroy like the binding sites for translocase, like it can't bind there anymore. And so what happens is like you're inhibiting like the whole like translocation process of which you're like moving the mRNA along the ribosome. And so it acts as an enzyme as well. Like RTA acts as an enzyme, which makes it so toxic. So like this would probably be like the main bullet point in terms of like this slide about like ricin and how it works is like, that's why it's like so toxic. And so lastly, I just want to quickly go over post-translational modifications. So one of them is proteolytic processing. So this is something that we were just covering a lot of in biochem in general, but specifically for this, for translation, obviously we have formylmethionine. So this is just, this isn't bacteria only, right? So formylmethionine is going to be that first one. In order to get rid of that formal group, we have something called peptide deformylase. We get rid of that peptide. We get rid of that formal group, and we have now methionine. And methionine is going to be cut by something called methionine aminopeptidase. So it's important to know like the name of these enzymes, specifically this one, because this one is going to be like really popular for cleaving like both prokaryotes and eukaryotes, like the methionine, which is stemming from like that AUG start codon. And so once that's cleaved, like that's basically what happens is most of our proteins essentially like are going to be cleaved by like methionine aminopeptidase. It's going to get remove that first one. And now in terms of this, like this is kind of like a review of just like what Purich already went over. I know it's been a while, but basically the main point is that zymogens are inactive. The reason why we store our enzymes in terms of the inactive state is that we don't want like unwanted reactions and like, you know, like a degradation of just random stuff, or like unwanted reactions essentially. So we have like trypsinogen, and that's going to be inactive. And trypsinogen can be, turn into trypsin, which is like the active form by enteropeptidase. And enteropeptidase is just another example of just proteolytic processing because it basically cuts out like the first 6 amino acids in order to activate your trypsinogen and turn it into trypsin. So now lastly, in terms of post-translational modifications, there's covalent modifications, but the thing is like, I don't think you guys really have to know in detail like what's going on, but you should know like the things in bold and like know that they're examples of modifications. Like you have phosphorylation by pKa, which you guys are like well like known, and like you guys know that because of like Xylee and his lectures. And then you have like gamma carboxylation, which I believe Purich covered, but that's basically of glutamate. And then you'll have like methylation. So methylation has to do with obviously turning off genes, and Stephen mentioned this like and was talking about like methylation, acetylation, and stuff like that. But methylation has to do with basically turning it off, and acetylation has to do with turning them on in terms of like the histones. And this like regulates the chromatin structure if you have like an open or more of like a compact closed chromatin structure. And then also most of like our proteins are going to be N-acetylated by something called N-terminal acetyltransferase. And what happens is like we cut the methionine out, right? But we also, that N-terminal right there can be acetylated. And so that's what happens by this enzyme. And lastly, so I guess like the main one in terms of this would be like the ubiquitin and protein turnover. So you guys have learned about protein turnover and how like if we have unwanted proteins, like we'll basically mark them for degradation, right? Because they're unwanted, or if you have damaged proteins or stuff like that. So you have like ubiquitin right here, and you have something called E1. E1 is going to be like your activating enzyme, and it's going to have a cysteine residue. So we're actually going to bind ubiquitin to the cysteine residue. It's going to bind it's going to basically, E1 is going to attach ubiquitin to itself, and it's going to attach it to here, and then it's going to transfer it to E2, right? And it's going to transfer it onto E2 cysteine residue. And then E2 is going to transfer it onto a target protein. And specifically, it's going to bind to a lysine residue, right? The ubiquitin. So we're basically transferring ubiquitin as we go from E1 to E2 to E3. And E3 is going to be a ligase. So what it does is the ubiquitin is going to be transferred by the ligase to the lysine residue, and it's going to mark the protein for degradation. But the thing is, not like one ubiquitin molecule won't mark it for degradation. You need to have like a whole chain. And I believe this was like on my exam, is like you need to have like one of the trick questions was like you had to have the whole chain. Like it's not specifically just ubiquitin that marks it for degradation. You need to have like multiple or polyubiquitin, which it says like right here in red. But essentially the like way we get polyubiquitin is like this ubiquitin has lysine residues. And so we obviously want to bind ubiquitin, like free floating ubiquitin, to these lysine residues. And so what the ligase does is it takes these ubiquitins and just attaches it to itself, because these have like lysine residues in it. So it just keeps attaching it, and like we have a whole chain essentially. And so the process just keeps continuing until like, you have polyubiquitin, and then it just marks it for degradation. Yeah. And then this is lastly just a slide of just comparison. This is like an example of something that you guys could do that would really help. I feel like would help for like understanding the processes. This is just for translation, but like the different stages, just like a comparison of the two. And I recommend doing this for like, replication and transcription and all the other processes that you guys learned. But yeah, I think that is it. So I'll pass it on to the next presenter, but I just wanted to say thank you guys for attending. It's great, and I appreciate all of you guys for attending, and like hopefully these sessions are really helpful, and I wish you the best of luck on exam 4.
Hey, Kashan, can you? Oh, yeah. All right. So I'm gonna be going over his lectures 49 and 50. Can everybody see my slides or my screen at least? Yeah. All right. Perfect. All right. So we're gonna be going over his lectures 49 and 50. So lecture 49, we talked about DNA damage and repair. So the first thing we talked about for that was our Ames test. So our Ames test is basically one test that we can determine if any chemicals are carcinogens. Okay. So if we see here right on the side, if there's a possible mutagen, we're going to have more mutant causing colonies than if we didn't. Okay, so this is just representing like, okay, we have a lot of mutant colonies being caused by this possible mutagen when compared to the control. So the Ames test, I don't remember showing up on my exam, but it's just good to be aware of the purpose of the Ames test, which is to determine if any chemical or any if any substance or anything is carcinogen. Okay, so just understand that Ames test is used to determine that. So jumping into the types of damage of DNA, we have deamination, which usually converts oh my God, sorry, I forgot the name right now, but we usually just convert one nucleotide base to the other, and it's sometimes spontaneous. In other cases, for example, with our adenine and thymine, they occur spontaneously, but they're really slow. So sometimes we don't think of them as being a deamination type of DNA damage. But we do have chemicals that allow this to happen more frequently and more accelerated. So our sodium nitrites, our sodium nitrate, and our nitrous amine can usually accelerate the rates of deamination. For oxidation of DNA, our reactive oxygen species, our ROS, can actually cause an insert of a hydroxide free radical into our guanine and thymine. So this is really, really bad, right? And remember that we talked about our reactive oxygen species during Xyle's lectures and how we have a system ready for that to try to prevent some of those oxidation damages. And you can see why, because if we damage our DNA, it's pretty bad. For our depurination, this is caused by a hydrolysis of our NB glycosyl bond linking a purine base to the sugar phosphate, and it yields this abasic site. And be aware, this abasic site is going to come up again, but in terms of our DNA repair mechanism, okay? So other types of DNA damage is our spontaneous methylation. We can also have our alkylating agents as well. So these usually distort our double helix. So one way that we can think about this alkylating agents are our sulfur mustard gases. They bind to guanines in the same DNA and they distort the DNA completely. So thymine dimers, which I don't know why, but it's my favorite type of DNA damage. So this is caused by the UV radiation, and this results in this really complex formation where the thymines are attached to each other, and they completely turn the DNA around and distort it in a way that we'd never see. And then the worst type of damage is our DNA strand breakages, and these are caused by radioactive rays, basically. So our cosmic rays, X-rays, and radioactive materials, and they can be either single or double strand, and usually double is the worst kind of DNA damage, right? So here's just a picture of all the types of DNA damage. Here's my favorite DNA damage here, the thymine dimers. But you see that they all distort the DNA in some way, either by taking off the amino group, adding a methyl group, or completely distorting the DNA like our thymine dimers and our cross-linked guanines, right? So now we're going to talk a little bit about repair mechanisms. So for the first repair mechanism we're going to talk about is our mismatch repair. And this is just correcting any base pair mismatches that occur, okay? So for Dr. Doom, we only talked about the prokaryote. There obviously is a eukaryote as well. So for the prokaryote, our mut-L and mut-S complex recognizes that mismatch. This is a protein complex. The mut-H, which is associated with the mut-L and mut-S, they identify which one is the oldest DNA strand. So which one's the parental strand, okay? And the way that mut-H can do this is figuring out which one's the most methylated. So the most methylated DNA is usually the oldest DNA. So that's why we usually go using like which one's the most methylated, so we know which one's going to be the one that we're going to base our repair from, right? Because the older DNA, the most likely it is the correct one, the correct version. So after we've identified which one's the parent strand, mut-H has an endonuclease which nicks the unmethylated strand. Okay, so the younger strand. Then nick can be either 5' or 3' of the error. Okay, so we can have it both ways. And then an exonuclease will digest from the nick through the mismatch, and then DNA polymerase 3 will fill the gap, and DNA ligase will seal the nick. Okay, so if we look at the image right here, we found a mismatch, right? We come in, mut-L and mut-S come in and start preparing for the repair. Mut-H finds which one is the older DNA strand. It starts bending the DNA in a way that we are able to cleave. Since mut-H has an endonuclease activity, we can cleave it, and then exonuclease starts eating away from that where we cleaved it, so we can get that air, and then DNA polymerase 3 and DNA ligase repairs that little area that we just cut out of the DNA, because this repair mechanism isn't necessarily gonna just go for the one area that's damaged. It's gonna try to make a big gap so it can repair everything at the same time, because it's hard to just repair one little area of the DNA. So since this was kind of like your first example, so you're gonna recognize that repair mechanisms usually have 4 steps. We recognize the lesion, we remove or excise the lesion, we resynthesize the DNA, and then we ligate those loose ends. So this is just here the image of another way of seeing the mismatch repair. It's basically what I just said, right? So you see that the exonuclease doesn't only get that one area that has that mismatch repair, it just eats away like a big chunk, and inside that big chunk is going to be the damaged area that we originally wanted to correct. So now we're going to talk about our base excision repair. So this is just repairing mismatches resulting from a damaged DNA. So if you remember, mismatch is due to like replication errors, a bunch of like normal errors that we have. This one is due to actual DNA damages, right? So damaged base, depurination, and single strand breaks. So our DNA glycolase will cleave the NB glycosyl bond, making that AP site, that a basic site, right? We just talked about. This AP endonuclease initiates repair of the AP site by making a single strand break in the DNA at that AP site, and then DNA polymerase 1 that has that 5' to 3' exonuclease and a DNA synthesis activity will serve to resynthesize the DNA, and then DNA ligase will just repair everything else, right? So again, it's the same kind of steps. We find the damage, we exercise the nick, we resynthesize, then we ligate, right? So I honestly really like this diagram because I think it explains pretty much really well, right? So our DNA glycolase, and remember one thing that a lot, especially for eukaryotes, we have a lot of these DNA glycolase for each base, and sometimes multiple for the same base. Okay, so you see here that we have this uracil glycolase. So it's just going to remove the base, and then AP endonuclease is going to destroy that phosphodite or that phosphate DNA backbone. Okay. Once we have that phosphate DNA backbone, we're going to be able to use an exonuclease to, eat up this one area here, our 5' to 3' exonuclease. And then our DNA polymerase will resynthesize that huge gap that we formed, and DNA ligase will just reattach everything together. So it's always that same step, right? Okay. So now we're going to talk about nucleotide excision repair. So it's almost very similar to the same steps for prokaryotes and eukaryotes because it doesn't change that much, just the nomenclature. So the nucleotide excision repair repairs lesions that destroy the DNA double helix. So these usually come in the form of thymine dimers and those alkylations that we talked about. So for prokaryotes, we have our UVRABC exonuclease. They make two nicks in the damaged strand flanking the lesion, and then UVRD helicase will remove that damaged DNA, leaving a gap, and then DNA polymerase 1 fills the gap, and DNA ligase seals the nick. Same steps as usual, right? For eukaryotes, again, it's mostly the nomenclature that's completely different. So we have our XP exonuclease, which is this huge DNA complex, and it contains XPA through G, these subunits, plus our TF2H helicase that we talked about a couple of lectures ago, right? So what it does, XP complex scans for kinks, opens DNAs, and nicks the damaged strand. So it basically does what UVR ABC and UVRD helicase does in one single protein complex, right? So then the TF2H exercise that DNA, leaving that gap, and then DNA polymerase epsilon fills the gap, and then DNA ligase seals the nick. Again, I'll repeat it. It's always that same step, but sometimes it just changes the name of the protein, right? So here's just an image of what it does. You see that right in the beginning that we had this DNA lesion, right? That could be caused by thymine dimer, alkylation, whatever it is. But we're always going to have that same step of just removing a big chunk of the DNA where the damage is in, resynthesize, and then ligate. Okay, so more mechanisms. So these are going to be the ones that are going to become really, really important because they're really dangerous if we have some of these damages, right? So one of our direct repair mechanisms is the repair of the 0,6-methylguanine. And this is needed because it base pairs with thymine specifically. So, and then we have this MGMT, which fixes it directly, okay? It fixes any damages to the DNA directly. So this is very specific, or very different, from the other repair mechanism we talked about, because this one doesn't need to necessarily excise anything. It just immediately fixes it directly. So then we have our single strand annealing repair. So this is the image we see right here on the side. So this repairs double strand breaks. And again, double strand breaks are really dangerous. Sorry, I don't know what's going on with my throat right now. But we repair these double strand breaks, and the way we do it is that we have these exonucleases. So these yellow things represent complementary base pairings for the DNA. So we use our 5' to 3' exonuclease to digest the broken ends. So right, we just broke the DNA. We digest broken ends, and then single strand annealing proteins bind to repeated sequences. So what these yellows are going to be representing, right? Then our RAD52 facilitates base pairing between these homologous sequences. So we're trying to get those DNA strands back together by having those complementary base pairs right next to each other so that we can start, forming the DNA back into it, right? We have these flaps still going on, right? Because we had extra nucleotides on each side. So then we can actually degrade these flaps by our ERCC1 XPF endonuclease. And after we remove those flaps, we can use DNA ligase to seal the nicks. But if you saw, right, we just lost a huge chunk of DNA because of these endonucleases that destroy those flaps. So it is a type of an error-prone repair mechanism. Even though we do create an error in the DNA, it's more important to preserve the integrity of the DNA as a whole than it is to lose a couple of nucleotides. Other forms of these double strand break repair mechanisms are non-homologous end joinings. So these occurs in the G0 and G1 phases that we will talk about next lecture, what these are. And they repair also double strand breaks in mammalian cells. We have our Q7080 complex, which binds to the loose ends of DNA. CS is a kinase that initiates those bridgings of those broken ends. And then other factors bind and process those loose ends to produce sticky ends. Remember, sticky ends are what we allow us to bind like DNA strands together, right? Then we have our DNA polymerase mu and DNA polymerase gamma, which adds bases. And then our Artemis endonuclease removes those overhanging flops. And then, as usual, DNA ligase seals those nicks. Yeah, if you want to think about DNA ligase as the one constant throughout every repair mechanism. And then our homologous recombination DNA repair mechanism occurs usually in the S phase and G2 phase because the sister chromatids can actually be used as a template for the resynthesis of DNA. And they can also repair a double strand break in DNA as well. So if you've been aware, these three right here, single strand, non-homologous, and homologous are all going to be single strand and non-homologous are going to be error-prone repair mechanisms. So they will create certain errors, but our homologous recombination DNA repair is actually an error-free repair mechanism. So just be aware of that. So finally, we're going to talk a little bit about CRISPR and Cas9. So this is a huge a protein complex that was originally found in bacteria because this is actually used in the immune system for bacteria, but we can actually reconvert it for genome editing. So what we need for this is we need our CRISPR, which are clustered regularly interspaced short palindromic repeats. Big word or big phrase, I mean. Then we have our Cas9 endonuclease and our single guide RNAs or sgRNAs. And these contain these Cas binding sites. And they actually define the genomic target that we're looking at because they can actually base pair with it, okay? So the steps are that we have our Cas9 and sgRNA genes within a plasmid that are inserted into cells. The cells eventually produce those Cas9s and sgRNAs. Then our CRISPR-Cas9 complex searches for that target DNA because our sgRNA has those base pairs of the area of the DNA that we specifically want. And then the Cas9, since sgRNA is bound to the Cas9 can start making these double or single strand breaks. So double strand is repaired by non-homologous end joining. So it eventually does cause random mutation. So we can't really control it that well. But for single strand breaks, we can actually do a homology-directed repair. So where we can actually introduce a synthetic piece of DNA and then we can actually modify it the way we want it to be modified. Okay. So lecture 50. So lecture 50 is about signal transduction in the cell cycle. And honestly, this is my favorite lecture for the exam 4. Okay, so vocabulary, right? So we have hormones. So hormones are just molecules that are transported throughout the body. They come from a secretion cell and they move towards a target cell, okay? So these can be polypeptides, steroids, or amino acids. So they're usually active at very low concentrations and they're, they're subjected to a lot of regulation as we saw with Dr. Ziley's lectures, right? So we also have growth factors, which are signaling proteins that stimulate cell growth. And then these hormones and growth factors, for example, are going to bind to receptors that will cause, these receptors usually have really high affinities and specificities because they will eventually cause a signaling cascade, which will cause cells to do whatever they're signaled to do, right? So as I just said, the signal transduction, which is like a signaling cascade, is going to just alter the activity and it uses these secondary messengers. And you're going to see that we're going to have a very different cellular responses depending on what binds to the receptor, okay? So another thing for vocabulary is that you should know these types of signaling. So what an endocrine signaling is, these are the ones that we look at hormones. They travel throughout the bloodstream. Then we have paracrine signalings, which act on proximal cells. So paracrine is actually endocrine is going to be the most important one for this lecture specifically. We have autocrines, which is when a cell can actually act on itself. This is going to come into the cancer biology section when Danielle's going to talk about. And then we have juxtacrine signaling, which requires contact between cells. This is the one that we're not going to talk about as much. So these are the differences between a steroid and a protein or a peptide hormone. Steroids can actually diffuse through the cell membrane and then act on intracellular receptors, while hydrophilic signaling, so amino acids, some amino acids and peptides, they have to work at receptors from the outer plasma membrane to work, and they often work through a signaling cascade. So I know in like, I think 2 lectures ago or 2 slides ago, I mean, it says that we're not going to go through the mammary gland development, but I actually forgot to change that when we were using this slide. So we will talk about the mammary gland development because I think it's essential to understand everything. So mammary gland development, this is actually really important because it's a really good way of us observing the cell cycle and cell proliferation. So it's essential because it actually occurs postnatally, right? So it doesn't occur when you're, it's not finished once you're born. It will occur during puberty and during pregnancy. It kind of got blocked there, sorry. So the primary hormones that we're going to be using here are our ovarian hormones, our estrogens and progesterones, our growth hormones, and our insulin-like growth factors, okay? And I apologize for any of my students. I know you guys already seen the slides before, but bear with me. So there are 2 types of signal paths that we just said. So our peptides and our steroids, and these are good pictures to be observant of. So our peptides cause the signaling cascade, then we eventually can cause a cellular response. While our steroids have these intracellular steroid receptors, they can either be in the cytoplasm or even in the nucleus as a nuclear receptor. And these are going to cause a cellular response. So estrogen. Estrogen is very, very important for the development of breast and the mammary development. So estrogen is a steroid hormone, so it can cross the cell membrane. So when it crosses the cell membrane, we have these estrogen receptors on waiting for them, right? So in its inactive form, it's in this monomeric state, so it's by itself. Once estrogen binds, it actually induces a dimerization. So then we have 2 estrogen receptors and an estrogen molecule. And then this dimeric receptor will actually move into the nucleus. In the nucleus, we have something called our estrogen response element. And this is a cis-acting element, so we know that's inside the DNA, right? So with the estrogen receptors and estrogen complex, we can actually bind to the estrogen response element and activate a bunch of the things that are involved with whatever gene that has this estrogen response element. So estrogen receptor and estrogen complex will bind and bring in those hats, mediators, and chromatin remodeling complex. And for this case, specifically for the mammary development, we're going to end up getting our Jun gene. Okay. Remember, Jun gene is one part of the cell growth cycle. So we just got the first part. So how are we going to get the second part? So this is going to come in the form of IGF-1, so our insulin-like growth factors. Okay. Because Jun gene is not enough to fuel the mammary development completely. So IGF-1 is actually expressed when there's high levels of growth hormones and estrogens. And so once we have IGF-1, what do we do with it, right? We use it in the ROS-dependent pathway, okay? So the ROS-dependent pathway is a pretty big, important pathway. It has a picture right here, okay? So it binds to this receptor tyrosine kinase, causing autophosphorylation and dimerization, which activates the second messenger system. This autophosphorylation will also promote the association of the SHC GRB2 SOS1 complex. Then this complex will allow ROS to bind to GTP, I mean. ROS GTP will interact with ROF, which activates its kinase activity. Then ROF phosphorylates MEK, MEK phosphorylates ERK, and then ERK will phosphorylate whatever transcription factor we're going to be talking about. So this is just a diagram of what I just described. So as you see, binds to receptor tyrosine kinase, causes autophosphorylation specifically in this area right here. Then we're gonna have this complex, which will allow ROS to bind to GTP, which is its active form. ROS GTP will then activate the kinase activity of ROF. Then we're gonna have this phosphorylation cascade, if you want to call that. We're gonna go MEK, ERK, and then for this case specifically for our mammary development, we're gonna activate our ELK1 transcription factor. Okay. So ELK1 is a transcription factor, but specifically it's a transcription activator, right? Because it activates another gene. So once ELK1 moves to the nucleus, it's going to associate with something called a serum response factor. And once it has this complex together, it's going to bind to the serum response element. Think about it as similar to the estrogen and the estrogen receptor. They're going to have like similar nature to it, right? Once ELK1 binds to the serum response element, we're going to lead to the activation of FOS. And FOS is really, really important, right? Because this is the ultimate thing that we wanted. So now that we have Jun from estrogen and FOS from IGF-1, we're going to be able to form the Jun FOS, which is an activating protein 1. and this is named an early response gene. So AP1 is really, really important because I feel like it's like once you put your car or your keys in the ignition, like this is what's going to start everything else. So that's why it's called like an early response gene. And be aware, there are questions when they're going to say like what's an early response gene and what's a delayed response gene. So don't get mixed up with that because it's an easy point to lose, right? So AP1 again is important for the cell cycle. So once it binds to this area called the TRE in the DNA, it's going to induce the formation of MYC and cyclin D, which is a delayed response gene. MYC will come first. So once we have MYC, MYC can actually replace MAD. Okay, so MAD-MAX represses the pro-growth genes, but MYC can just go in there and squeeze in and just kick MAD out of that complex. Then we form the MYC-MAX, which actually will activate the pro-growth genes. Okay, so once we have these pro-growth genes, then we can actually start the cell cycle. So the cell cycle is pretty special because it's controlled by two different things. It's controlled by the cyclin-dependent kinases, or CDKs, and then the CDKs are in turn controlled by the cyclins, okay? So be aware of these differences, right? So cyclin D and E are present in G1, cyclin A is appeared in S phase, and cyclin B is in the mitosis, okay? So we're gonna mostly be talking about cyclins D, E, and A, okay? But just be aware cyclin B is a thing and it is involved in mitosis. So the upregulation of cyclin D, so the production of cyclin D by AP1 and MYC will actually start the whole cell cycle. So just imagine you moving your car after you just turn the ignition. And the important thing here is the cell cycle has two main commitment points. This one specifically is going to be really important, our restriction point for commitment to replication, which is between our G1 phase and our S phase. So this is what's going to be between those two barriers. We're going to have a lot of these CDK inhibitors. So our INK4 will bind to the CDK4 and 6, preventing its association with cyclin D. And our CAP will inhibit the activities of the intact CDK cyclin complexes, okay? So the inhibitors will be acting like a wall or a little hill. So only the increased stimulation of estrogen IGF-1, which again will lead to the AP1 and cyclin D, you know, that whole cascade of things will be able to overcome these inhibitors and move us towards the S phase. So, but let's go back a little bit and see what is actually happening in G1. So G1 is the domain of cyclin D and cyclin E, okay? So in G1, we're increasing our cyclin D CDK4 and our cyclin D CDK6, okay? But in between that, we have a repressor called our RBE12, which will inhibit pro-growth genes that are needed to enter the S phase. So what are we going to do with this, right? So our cyclin D CDK4/6 and our cyclin E CDK2 will actually phosphorylate that RB area or the RB part of that complex, which will dissociate, and then E2F can actually act as an activator, leading us towards that S phase. So once we're S phase, we need to start breaking cyclin D as fast as possible because cyclin D is actually going to inhibit DNA replication. And this is all pretty important, right? Because if we think about it, we want those cell cycle phases to be as distinct as possible, right? If we have let's say one area of the cell is at G1 phase and one area of the cell is at S phase, that's just going to, you know, that's spelling disaster. So as soon as we're able to get through G1 phase as a whole, you want cyclin D to start being broken down, and we break it down by them being ubiquitinated so that we can allow DNA replication to occur. So cyclin A is needed for the S phase, and cyclin A has a similar problem with a repressor called our SYK1. Good thing cyclin E CDK2 is still there from the G1 phase, which allows it to phosphorylate SYK1, and then SYK1 can actually be broken down, and then cyclin E CDK2 can actually promote DNA replication. So yeah, so I think that was everything. Again, I just want to repeat what Kashan just said. Thank you so much for letting us, you know, teach your review. Thank you so much for coming. We know you guys are going to do great in your exam 4. But yeah, that's it for me. Thank you so much. Sorry, Danielle.
You're all good, Matthew. Thanks, Matthew. All right. I am going to share my screen. All right. We are now in the home stretch of our review tonight. So I'm going to be covering lectures 51 and 52 in one final swoop. This, of course, is going to be covering cancer biology. So let's get right into it. This is just a brief overview of a lot of vocabulary that you're going to see coming up in the following slides. It's important to know that cancer tends to develop in tissues that require regular replenishment or have high rates of cellular turnover, because very often these cells which display this trait of high cellular turnover will have less restriction in terms of like things that inhibit them in order to replicate. So higher rates of replication. Some examples of these tissues within our own bodies are our skin, our blood, the lining of our digestive tract. These are a lot of tissues that will replenish very often. So following that theme of high cellular turnover, this happens very often in the epithelium of our organs. So cancer is most likely to develop in the epithelium of our tissues. These cancers of the epithelium are called specifically carcinomas. Specifically, a glandular epithelium cancer is called an adenocarcinoma. Some other types of cancer, which Dr. Duma covered, were leukemias, lymphomas, sarcomas. Yes. Tumor profiling is a term that was just mentioned. So essentially, in tumor profiling, we are taking samples of expression of cancer cells and sampling the levels of expression for certain cancer-related genes. A proto-oncogene is a regular normal cellular gene that encodes a protein which has the capability of promoting cell growth and proliferation. This is the normal cell gene, however, and then proto-oncogenes can be activated to function as oncogenes, which are essentially that mutant form of the proto-oncogene. This is a gene that favors excess growth. Specifically with oncogenes, you really only need a mutation of one allele for the oncogene in order to induce this accelerated growth. Like I mentioned before, activation is simply the process of a mutation that will convert a proto-oncogene into an oncogene. A tumor suppressor gene is a gene that encodes a protein which functions to inhibit cell growth and proliferation. In order to have a knockout of a tumor suppressor gene, it requires that we have both alleles mutated. So that's the difference to really emphasize there, is that in tumor suppressor genes, both of the alleles must be mutated in order to experience a loss of control. However, in these proto-oncogenes, it's really only one allele that needs to be mutated in order to lose control of cellular division. Dr. Duma also mentioned some cancer diagnostics during her slides. I just wanted to briefly mention a few of these. Several of the non-invasive cancer diagnostics which she discussed were sonograms, x-rays, MRI scans, which are especially utilized in detecting brain tumors, CT scans, PET scans, which are scans that essentially measure levels of fluorodeoxyglucose within the body in order to determine cells which are highly respiring. She did mention one form of invasive cancer diagnostics. She mentioned specifically the needle biopsy. Essentially, after the needle biopsy is performed on the tumor, it is looked at by a pathologist. Several indicators that pathologists will look for in order to determine if the sample is cancerous or not are high nucleus to cytoplasm ratio in terms of size, evidence of excessive rates of mitosis, so high rates of cellular division, prominent nucleoli, and also a loss of specialized structure. As I'll cover later in these slides, we find very often that tumors experience a loss of differentiation, a loss of specialization, and are in a more general and basic form of the cell type. So a very important question to ask ourselves when we're learning about cancer biology is, why does cancer happen? The most simple answer that I can give everybody is that we are multicellular beings, and this is the kind of the nature of being a multicellular organism. So the longer we live, the more likely we are to develop cancer, because essentially what causes cancer is an accumulation of mutations during our lifetime. So just in terms of probability, the longer you live, the more likely you are to accumulate mutations that could possibly lead to the growth of a tumor. Several of these mutations that I'm talking about can stem from errors in normal cellular functions, such as chromosome segregation during mitosis, replication of the genome. We can also experience spontaneous DNA damage. We can experience DNA damage from our environment. Specific environmental factors that can damage our genomes are called carcinogens. Radiation can also damage our genome, as we covered in several of the repair mechanisms before. These are termed specifically risk factors. Our environment can cause sporadic cancer, which is where a tumor arises entirely because of somatic mutations. And the important thing to understand here is that these sporadic cancers are cancers which arise from things that we did not inherit from our parents. They're in our somatic cells. Essentially, it's not heritable to your children. It's not something you picked up from your parents. It's purely a matter of chance and circumstance in your environment. You can also inherit a mutation from your parents. This is also a risk factor. If only one defective allele is inherited from your parents, you still will need that second allele to be damaged in order for the likelihood of cancer to really increase. But that person with the first loss of an allele is still at a disadvantage, as we'll cover very soon with retinoblastoma. All right, moving forward, I'm going to discuss the RAS-dependent pathways and HER2 gene with regards to loss of cellular growth control. So these are cellular signaling pathways that do govern cell growth. As we covered in the previous lectures, these involve receptor tyrosine kinases, which will react in response to a growth factor and therefore induce a cellular response, in this case, a phosphorylation cascade, which will then alter gene expression, signaling certain things to be produced within the cell, indicating that the cell needs to move forward in mitosis. So specifically with regard to these RAS-dependent pathways, all of these proteins that you will see here in the signaling cascade are correlated to genes that encode proteins in the pathway. So essentially the genes that encode these proteins are proto-oncogenes because they are the regular form of a gene which when mutated can possibly induce a loss of control within the cell replication. So gain of function mutations in these genes generate an oncogene. A gain of function mutation, specifically in the HER2 pathway, will result in the dimerization of the HER2 protein, even in the absence of the growth factor. So this gain of function mutation yields a hyperactive protein. I don't know if this terminology was exactly used in this class, but in a separate genetics class, which I took, we always associated gain of function mutations kind of like monkey wrenches in the cell. They still produce a protein. However, they produce a protein that may do things that it wasn't supposed to do or possibly be hyperactive. So it can mess up certain signaling pathways within the cell. Specifically with this gain of function monkey wrench HER2 protein, this is encoding a hyperactive receptor tyrosine kinase. The specific mutation that I'm talking about is a missense mutation that switches, essentially it's a mutation of a glutamine for valine, and essentially this yields a protein that favors receptor dimerization without the presence of a growth factor. So even in the absence of the growth factor, the HER2 receptor is going to phosphorylate itself, therefore activating this RAS-dependent pathway, essentially triggering a ligand-independent protein, which will stimulate the RAS-dependent pathway without the stimulation of a growth factor. And this pathway, specifically with regard to the HER2 gene, can be inhibited by what substance? You can shout it out or put it in the chat.
Herceptin.
Thank you. Yes. All right. So inhibiting activation of the HER2 gene. So Herceptin is a synthetic antibody that will bind to this mutated HER2 protein and prevent its dimerization. One important thing to note here is that even when the HER2 protein is being not allowed to dimerize, it is also being functioning essentially as a signal for a killer T cell to attach on to that cell that is experiencing this mutated form of HER2 and essentially kill the cell. So that's kind of a very intelligent logic behind using a synthetic antibody here, is that not only is it preventing this incorrect signaling, it is also killing the cell, which is expressing this, this broken version of the HER2 protein. So 25% of all breast tumors have HER2 overexpression. Herceptin is also widely used in combination with other chemo drugs. So it may not be a standalone treatment. However, it can be paired with chemotherapy in especially treatment of breast tumors. Next, I'm going to cover RAS mutations. Once again, a gain of function mutation in the RAS gene can result in a RAS protein that will interact with RAF, even in the absence of growth factor and interaction with this SOS1 protein. Like Matthew covered earlier in the cellular signaling, essentially RAS is a GTPase protein that when it is bound to GDP, it is in the off conformation, but once it is bound to GTP, it is going to set off a phosphorylation cascade. So this protein specifically phosphorylates RAF. And so even when this RAS protein is not told to start this phosphorylation cascade, it is going to do it anyway, which naturally can cause problems because we are essentially starting this cascade without signaling to start the cascade. Dr. Duma mentioned that HRAS, KRAS, and NRAS are all parts of the RAS gene family. Specifically with RAS mutations, they often mutate on the codon G12V. And like I said before, this results in a protein that cannot hydrolyze GTP. So this RAS protein is going to be stuck in the on conformation. So constantly signaling and being active, setting off that RAS dependent cascade. The protein that comes next in the cascade is RAF. We can also have gain of function mutations in our RAF protein. So a gain of function mutation in the RAF protein will result in a hyperactivity of the RAF protein kinase, even in the absence of growth factor. So you'll see in the diagram on the right that our receptor tyrosine kinase is not bound to the growth factor. Therefore, it's not phosphorylating itself. It's not associating with SHIC and GRUB2 and SOS. Therefore, RAS isn't even activated in this instance. However, because we have a mutated form of our RAF protein, it is still going to phosphorylate MEK and therefore set off the signaling cascade even in the absence of growth factor. This oncogene and gain of function mutation is specifically seen very commonly in melanoma. And Dr. Duma also just mentioned that the V599E is the most common mutation. I'm not terribly sure how important memorizing this number is, however. We also learned in our cancer biology section that chromosomal rearrangements are found very commonly in cancer cells. Chromosomal errors in chromosomal rearrangement can lead to aneuploidy, which is defined as an abnormal number of chromosomes. So if a cell is an aneuploid, it can have possibly 1 chromosome or 3 chromosomes. Essentially, it is just not a diploid. This can also include deletions, duplications, inversions, insertions, and translocations between diploid chromosomes, which will possibly be causing cancer. Chromosomal rearrangements are also very characteristic of medulloblastoma. One example of a chromosomal rearrangement which can cause cancer is the Philadelphia chromosome. When we have the Philadelphia chromosome, essentially what happens is that we have a swap of genetic information between chromosome 9 and chromosome 22. And when we experience this swap of genetic information, we are actually fusing the ABL gene on chromosome 9 to the BCR gene on chromosome 22. The result here is not a knockout of both proteins. It actually results in a fusion protein. When the ABL and BCR genes are combined on the same chromosome, they actually operate under the same operon, the same promoter. So essentially, we are synthesizing a combination protein of the BCR exon and the ABL exon. So this fusion protein has a hyperactive tyrosine kinase activity. You can see the fusion protein on the right here. And essentially, because we have this monkey wrench protein, it can have some pretty few consequences within our cells. We also cover the concept of the two-hit hypothesis. The two-hit hypothesis postulates that the inactivation of a single copy of a tumor suppressor does not result in a loss of growth control. Essentially, what we are being told here is that the loss of one allele of a tumor suppressor gene will not necessarily cause cancer. We can have a defective tumor suppressor gene that can be inherited. So this inheritance of one faulty allele can be the first hit of our two hits. And then subsequently, as we get older, as we develop defects in chromosome segregation, mitotic recombination, or a mutation, things that are especially likely during retinal development can result in a loss of the second and only functional allele. This is the second hit. This is also defined as a loss of heterozygosity. So once we have this second hit, we are actually homozygous for two faulty, two mutated tumor suppressor genes. The fact that chromosome segregation, mitotic recombination, and mutation are very common in retinal development is one of the reasons why familial retinoblastoma usually results in tumors of both eyes and a predisposition to sarcoma. We also in this course discuss the importance of the p53 tumor suppressor gene. This is one of the most important genes in terms of tumor suppressor genes within our body. The p53 protein itself is a zinc finger transcription factor, and it is something that our cell activates in response to cell stress, especially DNA damage. This transcription factor regulates multiple genes which are important for this response to cell stress. It can regulate DNA repair. It can sustain cell cycle arrest, so it can pause our cells within G1 and G2. And most importantly, it can be a signal for apoptosis, or a signal for the cell to kill itself. And essentially, in cancers, we find that this p53 gene is mutated in about 50% of all cases. So this naturally must be a very important protein in limiting tumor growth within our bodies if this mutation is found in half of human cancers. A mutation of the p53 gene is a loss of function mutation. Essentially, it will have a defective response to DNA damage, therefore increasing the mutation rate and chromosomal instability because the p53 gene is no longer able to signal for DNA repair, no longer able to arrest the cell in its mitotic process, and no longer able to signal for apoptosis. So naturally, because we are allowing the cells to operate with damaged DNA, this will not only increase mutation rate, but also jeopardize the stability of the chromosomes. So the first hit mutation for the gene that encodes for p53 can be caused by deletion, frame shift, nonsense, some missense mutations, and especially epigenetic silencing. The second hit, which can also be, like I said before, labeled as a loss of heterozygosity, can result from mutations or epigenetic silencing. It's at this point in the lecture where we cover that certain tumor suppressor genes can be inactivated via epigenetic silencing, which will essentially convert our transcriptionally active euchromatin into heterochromatin. So in the diagram below, say, for example, if this is the p53 protein that we're encoding for here, we didn't even have a mutation within the p53 protein exon. It was simply the fact that the cell was downregulating expressing activity, which will bring that chromatin closer together. So RNA polymerase can't bind, and essentially that protein is no longer being expressed. So this is just an example of when we don't even have a mutation per se of the genetic code itself. It's simply that the gene is being downregulated allowing for the possibility of increased mutations in chromosomal instability. Also in this lecture, we cover the cancer-causing capabilities of the human papillomavirus, or HPV. HPV is a virus that essentially inactivates tumor suppressor proteins within our cells. The human papilloma virus will use our cell's own machinery to make an E7 and E6 protein, which have the capability to bind and sequester our RB and P53 proteins, essentially very important tumor suppressor proteins within our cells. So naturally, because we have a lessened amount of this P53 and RB functional proteins, our cells are going to be more predisposed to mutation and chromosomal instability. Human papillomavirus specifically is very prevalent with cervical cancer. So cervical cancer occurs in patients with a persistent infection of HPV very often. HPV itself is a DNA virus that can be cleared by the immune system. And because our immune system can clear it, naturally, we can create a vaccine. The current and most popular vaccine for HPV is Gardasil. It is a vaccine that is a mixture of virus-like particles consisting of the HPV capsid protein. So it's not the genetic material itself. So you are not capable of contracting HPV through receiving the vaccine. It's simply the capsid protein generated by recombinant DNA methods. So essentially, this vaccine is exposing our body to those capsid proteins so we can more easily identify it and kill the HPV virus and recognize it quicker. So hopefully we don't contract HPV. Specifically with regard to breast cancer, there is a genetic predisposition that exists in about 5 to 10% of all breast cancer cases. Defects are most commonly found either in the BRCA1 or BRCA2 gene. BRCA stands for essentially breast cancer, BRCA. That's how I use the memory trick to remember those. So both BRCA1 and BRCA2 are involved in double-strand break repair. BRCA2 is involved in the strand invasion aspect of homologous recombination. So if we're thinking of homologous recombination from the previous lectures, in homologous recombination, we are essentially using the homologous chromosome as a template to fix our broken chromosome. And so BRCA2 is a protein that aids in that strand invasion in order to make a copy of that homologous chromosome genetic material. So naturally, if we are having issues with strand invasion, we are going to not have very functional homologous recombination capabilities. So this is a very important protein in terms of DNA fixing processes within our cells. BRCA1 is a very researched protein. It affects the E3 ubiquitin ligase activity of the cell. It affects cell cycle checkpoint control, chromatin remodeling, regulation of transcription, and chromosomal segregation. So based off of the functions of these proteins, it's pretty clear to see that if we have a faulty BRCA1 or BRCA2 protein, our cells are going to be very predisposed to DNA damage with not much defense in order to fix this damage. So if DNA repair mechanisms are impaired, naturally mutations are going to accumulate more quickly, and essentially speeding up the accumulation of mutations is going to make us more predisposed to developing tumors. One example of tumor progression which we learn is the progression of colon cancer. So like many other cancers, colon cancer can result from an accumulation of genomic defects. Specifically with regard to colon cancer, certain mutations can correlate to certain stages in the tumor development within the colon. With the loss of APC, so APC is a tumor suppressor gene within our cells. With the loss of this gene, it can result in an abnormal growth, a polyp. This is called hyperplasia. At this point, the cancer is still benign. A larger polyp will occur with a KRAS mutation. At this stage, it is labeled as an adenoma. Further tumor growth will occur with the loss of the DCC gene. DCC stands for like deletion in colon cancer. It's essentially, again, a tumor suppressor gene, which is knocked out in most forms of colon cancer. At this point, this polyp is still benign. Then it gets more severe when we have a loss of P53 activity within those cancer cells. Essentially, a loss of P53 activity occurs very commonly around the time when the tumor becomes invasive. And at this point, it is a carcinoma. Additional mutations will result in the ability for some cells to separate from that basal membrane, especially in epithelial cancers, and travel to create secondary tumors within the body, either via the blood or the lymphatic system. And at this point in cancer proliferation, we are calling it metastatic carcinoma, metastatic specifically because it is capable and is spreading to secondary tissues. Here are several invasive behaviors and essentially characteristics that will lead to metastasis of the cancer. Normal epithelial cells within our bodies do not move. They stay attached to this basement membrane within our tissue. And if the cells are functioning regularly, when they detach from this basement membrane, it will trigger that epithelial cell to kill itself. So this is a process called anoikis. Essentially, how tumor cells can metastasize is they will develop a behavior allowing the cell to break apart from this basement membrane, allowing the cell to possibly move throughout the bloodstream or the lymphatic system, allowing this cancer cell to become mobile. So essentially, because the cell is no longer triggered to kill itself when it detaches, it is able to spread to other tissues. So tumor cells can either invade blood vessels or the lymphatic system, allowing them to move systematically throughout the body. Once the secondary tumor forms, it is considered a metastatic lesion. Breast cancer cells tend to metastasize to the bone marrow, liver, or brain, or lung. And especially with metastasis, there are some sites in our body which are more favorable to become locations of secondary metastatic lesions, because in certain tissues, we simply learned that certain microenvironments are more suitable for tumor growth than others. So these are characteristics that we find commonly in cancer cells, specifically metastatic cancer cells. So very commonly in cancer cells, we will see that they display a self-efficiency of growth signals. So essentially, we have an increase in autocrine signaling. The cell is signaling to itself to reproduce, essentially not responding to growth factor, however, responding to its own growth factor. It's not receiving signals from an external environment. It is signaling for itself to replicate. We will see a loss of sensitivity to anti-growth signals. So essentially, any anti-growth barriers within the cycle of mitosis can often be ignored through a loss of sensitivity to those signals. They can evade apoptosis, which just one example of this behavior, especially in carcinomas, are that anoikis when the cell detaches from the basement membrane. They can experience limitless replication. This is commonly seen with a activation of telomerase activity. A little bit of background behind this is essentially every time our cells replicate, we lose a little bit of our telomeres. And if we lose enough of our telomeres, we will eventually have vulnerable chromosomes, which can essentially signal for no more cell replication or apoptosis because of those unstable chromosomes. However, telomerase is an enzyme which will rebuild those telomeres, and in cancer cells, they are essentially able to replicate limitlessly because their telomeres are not becoming shorter with each cycle of replication. They also display tissue invasion characteristics, which is also the evasion of anoikis in order for epithelial cancers to spread to other tissues. And they also sustain angiogenesis, which I will talk about in the next slide. So the ability to promote angiogenesis is a really important characteristic of tumor cells, specifically because tumors could not grow beyond 1 to 2 millimeters if they are not receiving sufficient blood supply. So tumors, of course, like any other tissue in our body, will need a certain amount of nutrients in order to keep growing. So as they are growing rapidly, they make up for this greater need of nutrients by essentially promoting growth for their own blood supply. So this involves tumors activating blood vessel growth. So they signal to nearby blood vessels for them to proliferate and even grow in the direction of the tumor. So often tumors will produce and secrete various angiogenic growth factors, such as VEGF and FGF, which will cause nearby blood vessels to grow faster and especially in the direction of the tumor. So that is about everything for lectures 51 and 52. I wanted to thank you all for being here. Thank you for such a fantastic semester of biochem. I wish you the best on exam 4 and all of your future endeavors. So at this time, I believe we are going to take any questions.